Well, hello everybody for day 2 of biochemistry coming your way. Day 2 of 11 days, 11 sessions of biochemistry. You know, last night when I went to bed, I said to my wife, I said, "Oh, I don't know, man. I don't know if I got through with the students. I don't know if I was able to get to them to understand. Don't memorize, don't look for mnemonics, but try to understand. And I don't know if I was getting the point across." And so my wife said to me, "Get a life, get a life. If the students don't show up tonight, that means you did a bum job." Okay, so I'm looking over here, and fortunately, a lot of students are showing up. So, whew, I guess it wasn't too bad yesterday. So hopefully, I'm getting the point across. But my wife, she has nothing to do with science or medicine like that. She's a preschool teacher, and she's always setting me straight. Always just like a little kid. Like you gotta do this, you gotta do that. She's not here right now. She went shopping. Spending my money. But in any case, I'm here with you tonight. I'm gonna have a good time talking about molecular biology. Okay, now what we're gonna do now is what have we done in this first 4 hours of biochemistry? What we did was yesterday we talked about in chapter 1. We talked about what chromosomes look like and what they are, made up of DNA, and we talked about what DNA is compared to RNA, and we talked about the precursors used to make DNA and RNA. We talked about nucleotides, nucleosides, bases, things like that in chapter 1. In chapter 2, we talked about the important topic of DNA replication, and that had to do with what's important to know, 2 major enzymes, topoisomerase 2 and telomerase, because of the integration with pharmacology and pathology and micro stuff like that. So we talked about DNA repair in chapter 2, major diseases in pathology and DNA repair. Remember the issue of Lynch syndrome? Remember xeroderma pigmentosum? Remember the issue of what's a tumor suppressor gene? Is it good for you, bad for you? All that was discussed in chapter 2. Those points were very high yield. So we talked about that in chapter 2. Now in chapter 3, we started chapter 3 talking about transcription. So again, you got to know what the word means, transcription. Transcription means making RNA. And so in this chapter, we talked about prokaryotic transcription. And as we talked about prokaryotic transcription, I talked about 6 numbers, and I was going to compare those with what happens in eukaryotes. And so that's where we ended up yesterday. We talked about 4 of the 6 comparisons with prokaryotic transcription. Remember, in eukaryotes, there is no operon situation because every gene has its own promoter. And remember, the promoter is the binding site for RNA polymerase in bacteria as well as in eukaryotes. And then we talked about that there are exons and introns. That's coming up in the next minute or so, exons and introns. How does the body deal with exons and introns? And then we talked about termination of transcription in eukaryotes. Not important to know for board exams. So that's where we finished up yesterday. And we talked about, now we're on page 41 of the book. And on page 41 is this figure. It looks complicated, but it's not bad. But what we have at the top is a pre-messenger RNA. You don't call it, after RNA polymerase finishes making this RNA, you don't call it messenger RNA yet. It's called pre-messenger RNA, sometimes called a primary transcript. And notice what it has. It has 5' UTR that will not encode for protein. Remember, UTR, untranslated. You got to know what that word means, translation. Untranslated region. So meaning that this part of the RNA will not encode for protein. And then we have the reading frame with exons and introns, going to be discussed in just a minute. And then there's a 3' UTR that will not encode for protein, untranslated region. So every messenger RNA in both prokaryotes and eukaryotes have 5' UTR and they have 3' UTR. Okay, so that's where we're at. So that's where we finished off yesterday. Now item number 5 of 6 items to compare with what happens in prokaryotes. Remember, in prokaryotes, item number 5, no transcriptional processing. Well, just don't, now that word NO, just delete it, because there are transcriptional processing events. Matter of fact, there are 3 transcriptional processing events for the RNA in you and me, and all 3 of them high yield. Now, the first of the 3, again, look at the words. Processing of the transcript. You know RNA is a transcript. Process, how the RNA becomes processed. There are 3. And the first of the 3 transcriptional processing events is that there is an enzyme you don't have to know on the 5' end of the RNA being made. This enzyme will put a 7-methyl-G cap. It caps the 5' end of the RNA. Now, what's important to know about this step, the name of the enzyme, not important. But what is important to know, this enzyme is putting the 7-methyl-G cap on the RNA co-transcriptionally, meaning before RNA polymerase finishes, this other enzyme puts on the 7-methyl-G cap here. Again, so this is being done co-transcriptionally. Before RNA polymerase finishes, the cap is added. The first of 3 transcriptional processing events. The second of the 3 transcriptional processing events is there is another series of enzymes. Don't worry about the name of the enzymes, low yield. But what these enzymes do, they put a poly A tail onto the messenger RNA and on the 3' end. And because it's on the 3' end, this is post-transcriptionally. After RNA polymerase finishes, this poly A tail is added post-transcriptionally. And again, the name of the enzyme, don't worry about that. But all you need to know is the poly A tail is added co-transcriptionally, post-transcriptionally. And what's co-transcriptionally is a 7-methyl-G cap. The poly A tail is post-transcriptionally. The third and last of the transcriptional processing events also is post-transcriptionally, and it's a major transcriptional processing event, and that is the splicing. And this is being done post-transcriptionally. Now, what happens is there is a particle in the cytoplasm, and this particle is called a spliceosome. You might see it called snRNA, low yield, but this is the spliceosome. And what does a spliceosome do? It's a particle in the nucleus. And what it does, it looks at every intron. Now, not shown on this figure is on the 5' end of every intron, as shown by the green arrow. That's the 5' end of the intron. On the 5' end of every intron, it's what's known, not shown here, it's what's known as the 5' donor sequence. What that sequence is of RNA, not important. But every intron will have a 5' donor sequence, donor site. And on the 3' end of every intron, it's what's known as a 3' acceptor site. 5' donor site, 3' splice acceptor site. Every intron has that. Now, what does a spliceosome do? Don't worry about the details, but you must understand the concepts. Now, I use my trusty cord. Suppose this white cord represents an intron. And suppose this fist represents an exon, the other fist represents another exon. And in between these 2 exons is the intron. Now, as you look at it, here is the 5' splice donor sequence, and here is the 3' splice acceptor sequence. Now, what does a spliceosome do? What it does, it comes along and loops the intron and brings together the splice donor and acceptor sequence, and by bringing them together, they'll base pair. Then the spliceosome then scissors off the intron. That's sometimes referred to as a lariat, looping like a rope. You're looping out the intron, and therefore both exons have been spliced together. One more time. Exon, exon, intron. The spliceosome comes along and it sees the 5' splice donor sequence and brings it together with a 3' splice acceptor sequence and loops the intron. And then by doing so, it then scissors off the intron, and now both exons have been spliced together. And notice here in this diagram, now exon 1 has been spliced together with exon 2. Now if you're a thinker, and you are, you would say to yourself, man, this splicing has to happen not 99.9% perfectly. This has to happen 100% perfectly. Why? Because this is the reading frame. These nucleotides and these codons, this is the reading frame for a protein that's going to be made. That's the next chapter, chapter 4. But this is going to be the reading frame. If there was incorrect splicing, the reading frame can be messed up. And you would imagine, man, there's got to be, you would think, you would predict in pathology, there's got to be a disease that's due to incorrect splicing. There are many, many diseases that are due to incorrect splicing. And many diseases. I'll give you one great example you must know for board exams. Beta thalassemia. Now in beta thalassemia has 3 exons and 2 introns. The beta globin gene has 3 exons and 2 introns. And in intron 1 of the beta globin gene, there is a mutation in the splice donor site of intron 1 of beta globin. There's a mutation in the splice donor site. So as a result, the spliceosome doesn't see the 5' splice donor site in intron 1, and it doesn't remove intron 1. That is the cause of beta thalassemia. So if they ask you what's the molecular basis of beta thalassemia, you say there's a mutation in the splice donor site of intron 1, and the spliceosome cannot remove it. Many diseases, other examples, systemic lupus. In systemic lupus, there are antibodies against the spliceosome. Other great examples is Marfan syndrome. In Marfan's, there's mutations where the intron is not removed. Another great example is Tay-Sachs disease. Let me see your answer. Let me see. Now, in the book, they might talk about it, but in Tay-Sachs disease, what's the name of the enzyme? What's the name of the enzyme that gives rise to Tay-Sachs disease? You got to know Tay-Sachs for board exams for many, many reasons. What's the name of the enzyme? It's called hexosaminidase A, as therefore you end up with a defective protein. And then let me see your question. Let me see your answers here. What accumulates in Tay-Sachs disease? What's the name of the molecule that accumulates in Tay-Sachs? That's right, it's called GM2 ganglioside. Now, if you forgot that, don't worry. We'll talk about that later on next week and once we get to the sphingolipidosis. But the important feature here is that there are many diseases. Other examples, Gaucher's disease in another sphingolipidosis. There are many diseases that are due to incorrect splicing because it has to be done 100% perfectly. So another great example, you don't have to know this detail, but in type 2A, hyperlipidemia, it's a defect. Let me see your answers here. What is the name of the protein? If you forgot, don't worry, that's coming up next week. In type 2A, hyperlipidemia, what's the name of the protein that's defective? If you forgot, don't worry. That's all coming up next week. I'm just trying to see if you can recall. Type 2A hyperlipidemia, what's the name of the protein? No, good try. At least you're trying, Daniel. Take care. Good try. And the answer is, Leonardo, getting close. It's ApoB100 receptor, the LDL receptor. You got to use the word receptor, not ApoB100. It's the receptor for B100. And you don't have to know this level of detail, but the LDL receptor gene, when it's made into RNA, the LDL receptor RNA has something like 19 exons and 18 introns. That's staggering. 18 introns have to be removed perfectly, and then all of these exons are then spliced together in the right reading frame. And as you might imagine, in type 2A hyperlipidemia, People have lots of mutations because they can't splice the introns properly. Now you should ask why, the why question. Why do we have introns in our genes, in our RNA, if after all they're causing all these diseases? That's coming up. Stay tuned. It's coming up shortly. Why do you have introns in the first place? Because that's a logical question. You're a thinking person. You would say, well, why do you have all these introns if they're causing all kinds of pathology? Stay tuned. It's coming up shortly. All right, now to finish this off here. So the RNA that's being made undergoes 3 kinds of transcriptional processing events. And this is all going on in the nucleus. The 7-methyl-G-cap is added co-transcriptionally. The poly-A tail is added after the RNA is finished being made. And then the introns, after the RNA is made, the introns are removed in the nucleus. And then you can call it the messenger RNA. But before that, these RNAs are very heterogeneous in size. That's what the little H stands for, heterogeneous nuclear. That's where it's present. Heterogeneous nuclear RNA of different sizes because it's being processed. But after all this processing, then you can call it mature messenger RNA, which then leaves the nucleus and then gets out into the cytoplasm. And when it gets out into the cytoplasm, that's where the ribosomes are located in your cells and mine. And that's when the ribosomes jump on. And if you ask, let me see your answers here without looking. What's the name of the, how do the bacterial ribosomes know to jump on to the messenger RNA in prokaryotes? Don't look on that previous page. I'm trying to get you to recall. How do the bacterial ribosomes know to jump onto the messenger RNA? That's right, the Shine-Dalgarno sequence. Fumi, when you say promoter, that's RNA polymerase, not ribosomes. Please get those straightened out. But at least you're trying, Fumi. Keep trying, keep trying. The promoter is the binding site for RNA polymerase. In bacteria, the Shine-Dalgarno sequence is the binding site for the ribosome to carry out translation. In eukaryotes, it's the 7-methyl-G-cap that is the binding site for the ribosomes. That's how the ribosomes know to jump onto the messenger RNA and begin the process of translation, coming up on the next page. Please get these. That's high yield. Don't wait to master this material. You must master it as soon as you can. It's not hard. It's not hard. But why is it so important for boards? Because coming up a little bit later in the next chapter, I'm going to ask you, what's the name of the pharmacological drugs that are used, antimicrobial drugs that take advantage of your knowledge that in bacteria, the ribosomes jump onto the Shine-Dalgarno sequence? We don't have Shine-Dalgarno. That's why those drugs have no effect. Those antimicrobials have no effect on me. Otherwise, you're just going through the motions. If you don't understand, you must understand. You must understand concepts here. It's not hard. It's not hard. Give yourself credit. You can do this. It's just a matter of thinking. It starts to make sense. Okay, so the ribosomes will jump onto the 7-methyl-G-cap. They'll move on the messenger RNA until the start codon is encountered. That's coming up in the next chapter. And then the RNA is then translated to make a protein, starting with the amino terminus to end up with a carboxyl terminus when a stop codon in the next chapter is discussed. So, okay, that is the issue of translation here. Okay, now what's coming up next is the issue, major. They love this on board exams. Any board exams, whether it's U.S. boards or Canadian boards, Comlex or whatever, they love this next topic. And that is, in the body right now, why do you have all these introns in the first place? Why do you have them if they're causing all kinds of diseases, if they're the basis of many diseases? Why do you have all of these introns in the first place? Why can't we be like bacteria where there are no introns? And the reason is You must master the reason. And the reason is because in your body right now, excuse me, in your chromosomes, you have 20,000 genes. But in your cells, in your body, you have more than 20,000 different kinds of proteins. You have well over 100,000 different kinds of proteins. So the issue becomes, think about it, how can you have so many more proteins, different kinds of proteins in a cell, than you have genes in your chromosomes that can account for those proteins in the body? And there it is. This is a very high yield topic called alternative splicing. Now in alternative splicing, this is why you can have more proteins in the body than you have genes in your chromosomes, in your cells, that can account for those proteins. And this is high yield. I'll explain why it's so high yield. It integrates with other disciplines. That's why. But the idea is, let me give you the biochemistry first, the straightforward biochemistry. So at the top of the figure, suppose we have a primary transcript, pre-messenger RNA. Here is the 5' UTR. Here is exon 1, intron 1, exon 2, intron 2, exon 3, intron 3, exon 4, and 3' UTR. So this particular transcript has 4 exons and 3 introns. Now what can happen is that in the nucleus, the spliceosome might come along and take the splice donor site of intron 1 and loop it together with the splice acceptor site of intron 1, removing the entire intron 1 from the RNA, thereby bringing together exon 1 with exon 2. Nothing surprising, we've spliced exon 1 with exon 2. But what the spliceosome might also do, it might take the splice donor site of intron 2 and loop it with the splice acceptor site of intron 3. In other words, remove this entire region of the RNA. And by doing so, we now splice exon 2 with exon 4, no exon 3. And we'll call that messenger RNA version 1. Okay, everybody understand? Just simply by bringing together the splice donor site and splice acceptor site of different introns, we can loop out a region of the RNA. So we'll call that messenger RNA version 1. Now, what can also happen is the spliceosome might come along and take the splice donor site of intron 1 and loop it with the splice acceptor site of intron 2. Loop out this entire region of the RNA, thereby bringing together exon 1 with exon 3. No exon 2, because exon 1, exon 3 have been spliced together. And then the spliceosome might come along and take the splice donor site of intron 3, the splice acceptor site of intron 3, loop out intron 3, thereby bringing together exon 3 with exon 4. And now we'll call that messenger RNA version 2. Now think about this. Now what that would say to you, if you're a thinker and not a memorizer, if you're a thinker, you would say to yourself, well, how does this manifest? What's the significance of this? What is the meaning of this? What that would say to you is that the protein encoded by messenger RNA version 1 is going to be related to, but not identical with, the protein encoded by messenger RNA version 2. This is where you get different proteins coming from one gene. You can end up with these proteins will be related but not identical with each other. Here, they're going to be related but not identical. Now, why is this so high yield for board exams? Because of the integration with other disciplines. Now, you just had Dr. Muscatello, and so you should be able to answer this. In immunology, now don't look in the book, because the book starts talking about this, but don't look in the book, but just trying to recall from Dr. Muscatello's lectures. But in immunology, what's the best example of a set of proteins, different proteins that undergo alternative splicing that gives rise to different proteins coming from one gene? What's the best example of this? The best example. What about antibodies? What about, Vincent? What about antibodies? Heavy chains, well, no, heavy chains, light chains, that's a little bit different. Now, what this is about is this is where you can have the same immunoglobulin with the same epitope, the same IgG, for example, that's looking for the same antigen. But sometimes that immunoglobulin is membrane bound on the B cell, and sometimes that immunoglobulin is secreted into the bloodstream. And if the national board asks you, how does that happen? You say alternative splicing, where the RNA of the immunoglobulin is alternatively spliced. All right, now earlier you had Dr. Wilson. Now in Dr. Wilson's elegant lectures in physiology, how do you think this integrates with physiology? And anybody know how does this integrate with physiology? Or this alternative splicing. How might you see alternative splicing in molecular physiology? That's right, the triple myosin variants in the muscle from Dr. Wilson's lectures in physiology, muscle physiology. You got different triple myosin variants. If they ask you how does it happen, you say alternative splicing. All right, here comes. In endocrinology, that Dr. Wilson talked about. What's an example of a class of proteins, different proteins, class of proteins that are also generated by alternative splicing? That's right, a class of proteins generated by alternative splicing in endocrinology. And the answer is, it is the dopamine receptors in the brain. Remember those dopamine receptors that Dr. Wilson talked about? You got different kinds of dopamine receptors. If they ask you, how do you get these different kinds of receptor for dopamine? You answer, alternative splicing. Now let me say, you can see why this is high yield. Matter of fact, over 60% of your RNAs and mine, your messenger RNAs and mine, undergo alternative splicing. So this is a common event that goes in your cells, alternative splicing. So the national board expects you to know what it means. And matter of fact, later on tomorrow, I'll give you another example on board exams, how they might ask you about the concept of alternative splicing. Please be ready for it, because of all the integration. You're going to see it again in this alternative splicing tomorrow. Okay, so what have we done so far? We've talked about now in eukaryotes, we talked about messenger RNA, as well as in prokaryotes. Now to finish off the chapter, there are two other RNAs that we need to review. And one is tRNAs coming up on the next figure after this one, after the next figure, and then the next figure though will be ribosomal RNA. So we're going to talk about ribosomal RNA next, and then after that tRNAs. Now ribosomal RNAs. Now what you need to know about ribosomal RNAs, rRNAs. These are the characteristic most abundant of the RNAs. Remember, messenger RNAs are the most heterogeneous. You got big ones and small ones, all messenger RNAs of different sizes. But ribosomal RNAs, characteristically, are the most abundant. So if they ask you, which of the following RNAs do you have in a cell in greatest number? And it's the ribosomal RNAs. Now, the ribosomal RNAs are used to make ribosomal subunits. Now, in prokaryotes, there is a small subunit and there's a large subunit. What's important to know is these wavy line represents the ribosomal RNA complex with proteins. Don't worry about the details. I purposely took out the numbers here of the ribosomal RNAs. That's not important. Don't worry about the numbers. What numbers you should know, though, is the small subunit in prokaryotes is 30S. So the small subunit in prokaryotes is the 30S subunit, which is a complex of ribosomal RNA and protein. What's the protein? What's the ribosomal RNA? Not important. But what is important is it's the 30S subunit. Why is that so important? Pharmacology integration coming up a little bit later. Now the large subunit is the 50S subunit, complex of protein as well as ribosomal RNA. Now when they come together, 30S and 50S, now put away your calculator. because it won't work. 30S plus 50S does not equal 80S. It equals the 70S ribosome. So in E. coli, the entire ribosome is the 70S ribosome. Please keep that in mind. It's going to be important on the next chapter. So the entire so those numbers of the subunits are important. And I'll show you why. And I'll show you why in a little bit. In eukaryotes, and me and you, the small subunit is the 40S subunit. The large subunit is a 60S. And again, don't memorize these ribosomal RNAs, not important. But 40S and 60S together is the 80S ribosome. So not 100. Put away that calculator. It's not going to work. 40 plus 60 is not equal to 100. It's equal to the 80S ribosome. Don't worry about why. Not important. It's just the way you do ultracentrifugation. They're not going to ask that on board exams. But again, one more time. You should know 40 and the large is 60. Together in eukaryotes, it's the 80S ribosome. Now, why is that important? These ribosomal subunits. And one reason is because in prokaryotes, we talked about this already, that nothing new. It's the 30S subunit that recognizes the Shine-Dalgarno sequence in prokaryotes. And again, in a little bit, we're going to talk about pharmacology coming up in the next chapter. So you should know it's the 30S subunit that recognizes the Shine-Dalgarno sequence on the messenger RNA. In eukaryotes, it's the 40S subunit, the 40S, that recognizes what? Let me see your answers here. What does the 40S subunit recognize? That's right. It recognizes the 7-methyl-GCAP. So you must know that because pharmacology coming up a little bit later in the next chapter. I'll point that out to you. But another reason why you need to know these subunits, because the integration with microbiology. Now, in microbiology, you just had Dr. Muscatello when she talked about sugar toxin. Now, what happens is sugar toxin, why it can kill me and you. As sugar-like toxins or sugar toxin, E. coli has another toxin. This is the hemorrhagic E. coli has another toxin called virotoxin. So Shigella secretes sugar toxin, and hemorrhagic E. coli secretes virotoxin. And what they do is, now you see in the margin of your book, they cleave a ribosomal RNA in the large 60S subunit. I doubt they're going to ask you what's the name of the RNA, ribosomal RNA, but it is the 28S ribosomal RNA. But that's low yield. One more time. What's important to know is if they ask you, how does sugar kill? You answer. What sugar toxin does, it cleaves a ribosomal RNA in the large subunit of eukaryotic ribosomes. And because you're cleaving a ribosomal RNA, the ribosome doesn't work. And therefore, you will not make protein. And if you don't make protein, then death will show up. The Dr. Death will come knocking on the door if you can't make proteins. So the idea is, so that's all you need to know. Don't worry about the details, but if they ask you, how does sugar or virotoxin kill? We just talked about the idea. Okay. All right, now what's coming? So this is what you see right here in front of you on page 43 is all you need to know about ribosomal RNA right there, okay, in terms of the structure and stuff like that, okay. Okay, now what's coming up next, no reason to read about it, as long as you've been paying attention, as long as you're understanding, they got your back, just focus your attention on the high yield stuff we just talked about regarding ribosomal RNA. Now the last of the RNAs we need to talk about is tRNAs. Now tRNAs, a characteristic of them, these are the smallest of the RNAs. Remember, messenger RNAs are the most heterogeneous. You got big ones, small ones, processed, and all this kind of stuff. Ribosomal RNAs are the most abundant, and the tRNAs are the smallest, the tiniest, the very small RNAs. Now the idea, now all tRNAs look like this, a clover leaf structure, sometimes called a cross structure, a cross-like structure, but a clover leaf structure. But notice they have, here is the 5' end. They all wind around with these different loops all the way to the 3' end. All tRNAs have the same structure, but they can have different sequences, but all tRNAs have the same structure. Now, how many tRNAs are there in a cell? That's coming up in the next chapter. Don't worry about it now. But you have a lot of tRNAs in a cell. Now, I hear about tRNAs being on board exams many times in recent years, many times. And what a student would tell me, they'll take in the board exam and they press a button. On the monitor comes this cloverleaf structure. And generally speaking, what the students will say, they tell me they have arrows. The boards will have arrows pointing to the various parts of the molecule. And they ask, what's important about this part of the molecule or that part of the molecule? Okay, so here we go. So if the boards show you a tRNA structure, like you see right here. Now, first of all, what's the function of a tRNA? Before we talk about the various parts of the molecule, what's the function of a tRNA? Transfer RNA. It's picking up an amino acid and it transfers the amino acid onto a growing peptide, growing protein. So it picks up an amino acid to carry out protein synthesis. And the other function is to recognize a codon. If you're going to put an amino acid into a growing protein, you've got to recognize what's the codon. And this tRNA will recognize which codon we're talking about. Is it the codon for valine, isoleucine, leucine, something like that? Now, if you see a cloverleaf structure like this, and they have arrows pointing to the arms, these arms, and they ask you what's important about the arms, you say, that's where you have modified bases. Don't worry about the modification, that's not important. Notice over here, there's a base called Psi base, low yield. In fact, you can have some methylated uracils, and that's thymine. Remember all that yesterday's review? And so this is the only place in RNA you have thymine, is in the arms of tRNA. So these arms of the tRNAs can be modified. Now, if they ask you why are the bases on, some of the bases on the arms, why are they modified? Now, I was trying to think of an analogy to explain this. And to explain this is, as we'll talk about in the next chapter, you can view all this stuff we're going to talk about regarding messenger RNA, tRNAs, and ribosomes and all that kind of stuff coming up in the next chapter. You can envision all that stuff like a subway. You all been on subways, and so or metros or whatever you want to call it. If you live in London, it would be the underground or whatever. But again, all these metros or subway stations, subway trains. So the track, the railroad track, the train track, you can view that as messenger RNA. And the train, you can view it as the ribosome. So the ribosome is moving on the train track. So the ribosome is moving on the messenger RNA to make the protein. And so then one more thing is the tRNA. Now, the tRNA is bringing into the ribosome the respective amino acid. So you can view the tRNA as a passenger, a person. So again, if you've been on a subway train, just picture. So all of these people just view them as tRNAs. All these people are coming into the train, and the train is the ribosome. And then the ribosome is now moving on the train track. So that's the ribosome is moving on the messenger RNA. As the train moves, you got to hold on for dear life. And you got these things called arms. And the arms are going to hold on to hold on so you don't fall over. That's the same thing that happens here. As the tRNA comes into the ribosome, as the ribosome moves on the messenger RNA, the tRNA is holding on. And it's holding on using these arms. And they have the modified bases there. So there it is. Okay, hopefully you understand why they are modified. They use the arms to sit in the ribosome and not fall out. Okay. All right, now another item here. If they have an arrow pointing to the 3' end of the tRNA, what's important about the 3' end is that is where every tRNA, even though they may have a different sequence, all tRNAs on the 3' end have a CCA sequence. And on the A of the CCA sequence, that's where there is an amino acid that's in covalent linkage to the A. So here it is. So one more time. On the 3' end of every tRNA, whether it's a tRNA for valine, glutamate, aspartic acid, no matter what the tRNA is, they have a CCAN, and on the A, that's where the amino acid is in covalent linkage. Now let me stress here. Let me stress. You can imagine on board exams if they say to you, what is the sequence to which an amino acid is attached on a tRNA? What's the sequence? You can imagine option A is going to be CCA, option B is going to be ACC, just like you see here, ACC. Never say ACC. Why not? Let me see your answer here. Why don't you say ACC? That's right, that orientation. I hammered home yesterday that the orientation is 5' CCA to the 3' end. That orientation will mess you up if you're not paying attention here. All right, so now that's on the 3' end. And one last thing is, if they have an arrow pointing to the bottom of the molecule, and they ask you what's important about the bottom of the molecule, you say that's where there's an anticodon. An anticodon that's going to base pair with a codon in a complementary and anti-parallel fashion. The anticodon will be 5' CAU to the 3' end, and the codon in the messenger RNA will be 5' AUG to the 3' end. Notice how the anticodon-codon base pairing is complementary, C with the G, A with the U, U with the A, and anti-parallel. Always, always, always. So if they have an arrow pointing to the bottom of the molecule, and they say, why is that important? You say because of that's where you have an anticodon be involved in codon base pairing. Okay, everybody, we're done with chapter 3. Okay? Any questions on chapter 3? So again, if you have any questions, feel free to use the Q&A pod, and Dr. Seda, Dr. Donald will be happy to go through your answer for you, and if they feel it's important for me to know, then they'll send it off to me. But this is a very efficient way of doing this. Instead of pausing all the time, Dr. Seda, Dr. Donald on board tonight will be happy to answer your questions. Okay, so so we're done with chapter 3, and the next chapter is chapter 4, talking about translation. Again, chapter 1 dealt with chromosomes. What's chromosomes? Chapter 2 deals with replication. Chapter 3 deals with transcription. And now chapter 4 deals with translation. And before we talk about translation, we got to talk about the genetic code. And after that, we got to talk about mutations. And then we'll get into translation. This chapter is a very long chapter. It'll take several hours to go through this chapter. But be ready for a lot of high-yield integration with pharmacology, pathology coming up, microbiology coming up in this chapter. Extremely high yield. Okay, now regarding the genetic code, we've got to review the genetic code. And what the genetic code is, let me ask you this before we turn the page. How many codons are there? How many codons are there in the genetic code? How many codons are there? That's right. And the answer is 64. So there are 64 possible codons. In other words, excuse me. In other words, how many different combinations can you have of 4 nucleotides? How many different combinations can you have of 4 nucleotides in DNA? And the answer is you have 64 different combinations. And of the 64 possible codons, 61 actually encode for amino acids, and 3 of them do not. And they actually encode for the stop codons, we'll talk about in just a few minutes here. Okay, now, without looking at this figure here, don't look at the genetic code. Let me ask you another question. How many amino acids are there? If there's 64 possible codons and 61 actually encode for amino acids, how many amino acids are there? How many? You should know this answer. That's right, there are 20 amino acids. So what that means is that the genetic code sometimes is referred to as redundant, sometimes meaning degenerate. It's a redundant, degenerate genetic code, meaning what that means is you can have multiple codons specifying the same amino acid. Multiple codons, 61 different codons can actually encode for amino acid. 20 amino acids, you have multiple codons specifying the same amino acid. So what you see in this genetic code, here is the first position nucleotide of a codon, here's the second position nucleotide of a codon, and here's a third position nucleotide of a codon. Now the genetic code could be, usually is asked this way, where there's no work. They tell you what the genetic code specifies. Notice there U specifies phenylalanine, and so does UUC specifies phenylalanine. UUA specifies leucine, and so does UUG specifies leucine. So multiple codons specify the same amino acid. Down here, for example, 4 different codons specify glycine, and the difference usually is in the third position, sometimes referred to as the wobble position. But the idea is you can have multiple codons specifying the same amino acid. Now I want to stress, don't go memorizing the genetic code. Don't go memorizing that UUA specifies leucine. They can ask you to work it out by giving you the genetic code. In fact, on the next figure, we're going to use the genetic code. How might they ask you to use the genetic code? So don't go memorizing the genetic code. But in my opinion, if you could just remember 4 of the codons and what they specify, you save valuable time. You don't have to memorize them, but you will see the utility of knowing 4 codons. You save valuable time. And the first of these is the start codon. The start codon in any messenger RNA is AUG in both prokaryotes and in eukaryotes. And notice that AUG specifies methionine. So all proteins start with methionine in prokaryotes and in eukaryotes. Here, so they start. That's the start codon is AUG. Now let me ask you this. Suppose they give you a sequence of DNA. On the monitor in front of you, they give you a gene, a sequence of DNA, and it's 1 strand. And they say, here is a sequence of DNA, here is the genetic code, figure out the amino acid sequence. What do you want to look for on that DNA to begin looking at that DNA to figure out the amino acid sequence? What is the start codon? What's the start codon? What's the start sequence for a codon in that DNA? What's the start codon? Not U. DNA does not have U's. That's right. Now we're getting somewhere. That's right. It's a TG. So in other words, if they give you a sequence of DNA, remember, you must assume it's the 5' end on your left, and you must assume it's the coding strand. And if it's the coding strand, the DNA coding strand is identical to the messenger RNA, with the exception of T's and U's. So ATG in DNA means AUG in RNA. So if you could just remember the start codon, either in RNA or DNA, you save valuable time. Now the other 3 codons are the 3 stop codons, and they are UGA, the next one is UAA, and the other one is UAG. Notice over here in the genetic code, these are the stop codons. Now again, you don't have to memorize these, but if you could just remember them, you save valuable time. Now, some books like First Aid, they say you can memorize these 3 stop codons by little phrases like UGA. UGA is you go away. And UAA is you are away. And then UAG is what? UAG. Oh, that's right. Danish and Funmi, that's right. It is you are gone. Is there an easier way to remember these 3 stop codons? An easier way. And the way I remember these 3 is by the universities. Here it comes, the universities. UGA, the University of Georgia. UGA, the University of Georgia. The Bulldogs, they're good for something. UGA, they are a stop codon. UAA, University of Alaska at Anchorage. UAA, University of Alaska at Anchorage. Some of my friends who are in Alabama, they would say the University of Alabama at Auburn, or the University of Auburn at Alabama, but whatever. But University of Alaska at Anchorage, UAA. And then the last one here is UAG. Here comes the university, the Universidad Autónoma de Guadalajara. All of those of you who are from Mexico should get this one, UAG, Universidad Autónoma de Guadalajara. I've been there a number of times, that university. There's a medical school there, UAG. Beautiful weather in Guadalajara. Beautiful weather in that city. All right, so this is now the 3 stop codons. Now what we're going to do is we're going to take a 10-minute break. And then I'm going to come back, and then we're going to talk about the use of the genetic code. How do you use the genetic code? And the next figure, extremely high yield integration with pathology. Okay, everybody, I'm going to go off camera, and Donna will bring in the timer for 10 minutes. So see you in 10. Genetic code. Okay, now the use of the genetic code. How might they ask you to use it? And the way they could ask you to use it is a major figure coming up because of high yield integration with pathology, and that's this issue of mutations. So now on this figure, before I'm done with it, there's going to be a lot of color I'm going to be adding onto this figure. So you might want to write small because a lot of pathology coming up here. Now, suppose on board exams, they give you the sequence that you see right here in the upper part of the figure. And they say this is a normal sequence, and they say this is a normal sequence, and they give you the genetic code, and they say, figure out the amino acid sequence. So two assumptions you must make. You assume it's the 5' end on your left, and you assume it's a coding strand. Remember, nothing new. You had to be here yesterday to remember this. Now, since it's a coding strand, then you look for the start codon. When it's made into RNA, you look for the start codon in the gene called ATG. And to make life simple, ATG is right at the beginning of the gene used to make RNA. So ATG in DNA means AUG in RNA. AUG in RNA in the genetic code means methionine. GCA in DNA means GCA in RNA. That means alanine. ATT in DNA means AUU in RNA. You go back to the genetic code. AUU in RNA means isoleucine, and so on and so forth. Here. All right, now the one thing I want to mention here is that even though all proteins begin with methionine because of the ATG start codon in the gene, which transcribes the AUG in the RNA, because AUG means methionine in translation, it does not mean that all mature proteins have methionine at position 1, because methionine may be cleaved off. But they start the synthesis with methionine, but the methionine may be cleaved off while it's being made. Okay, all right, now we're going to focus our attention on this codon right here. Let me highlight it in a gray box right there, TTA. Now, TTA in DNA means UUA in RNA, and UUA in RNA, you go back to the genetic code, it means leucine. All right, now suppose the sequence at the top here, that's the normal DNA sequence making the normal protein. Okay, nothing strange. Now we're going to focus our attention on this codon, and specifically on position 3, the A. On position 3, suppose that A was mutated to a G, and all the other nucleotides, all the other codons are normal, but in this particular codon, instead of being TTA, it's now TTG, because that A was mutated to a G. Now, if you're thinking, what do you mean mutated to a G? It means in the germline of some individual, that when, in this gene, when the DNA polymerase was replicating this DNA, this gene, when it was replicating, you gotta know what the word means. When the DNA polymerase was replicating this gene, when it had this A here, it should have put an A in the newly synthesized DNA, but it made a mistake and put a G there. And normally, the DNA polymerase pauses and checks, like we talked about yesterday. And remember, the enzyme 3' exonuclease should take out the mispaired nucleotide because it gives rise to high fidelity. But as I mentioned yesterday, it's not perfect fidelity. Sometimes the DNA polymerase, when it makes a mistake, it doesn't pause and check and correct itself like it should. And so as a result, the A is converted to a G by mistake, and it was not corrected. And then the DNA polymerase proceeds and then puts in the other nucleotides and the other codons. So in other words, in this gene, all the nucleotides and codons are normal, except at this position, the codon is TTA, and now the codon is TTG. On the third position, nucleotide was changed. Now, TTG in DNA means UUG in RNA. You go back to the genetic code, and you will see that UUG also specifies leucine. So in the protein being made, there's a leucine where leucine should normally be. So is there any pathology in that individual? Absolutely not. And so this is what's referred to as a silent mutation. Silent mutation by definition is, please pay attention to this figure. A lot to say in this figure. A silent mutation is a single nucleotide change in the gene being made, which does not result in an amino acid change in the protein being made. So a silent mutation is a single nucleotide change which does not result in an amino acid change. So therefore the protein is normal, and this is what's referred to as silent mutation. So even though the gene has a mutation in it, there's nothing wrong with the protein being made. It's normal, the person is normal and healthy, no problem at all, even though the gene has a mutation in it. And usually these silent mutations are in the third position nucleotide. Again, sometimes called the wobble position, low yield, but usually in the third position. And the reason for and why you can have the same amino acid, even though there's a mutation, is because the genetic code is redundant. It's degenerate. Remember glycine? There are 4 different codons for glycine, and they all differ on that third nucleotide. So you can have a mutation in the third nucleotide for glycine. Ain't going to be a problem here. So silent mutations, no pathology. Now continuing on. Now suppose that the A in the third position was not mutated. It remained as an A in the third position of the codon. But suppose that T in the second position was mutated to a C. So the DNA polymerase, when it was replicating this gene, which is going to be passed on to the progeny of an individual, that T was converted to a C, and it was not proofread properly. It was not corrected properly. So C remains as a C. All the other nucleotides, all the other codons are normal, except at this position. The codon is, instead of being TTA for leucine, it's TCA, and that TCA for DNA is the same as UCA in RNA. You go back to the genetic code, and UCA specifies serine. So think about it. In the protein being made, there is a serine. Now, we haven't talked about the amino acids yet, but the amino acids have serine is a polar amino acid. It has a hydroxyl group, and leucine is hydrophobic. So in the protein being made, there's a serine, polar serine, or a hydrophobic leucine should be. So what do you think is going to happen to the protein being made? It's not going to fold properly. It won't fold. The protein likely will not work. The cell suffers. The body suffers. Pathology will then show up. And this is what's referred to as a missense mutation. So here comes the definition. A missense mutation is a 1 nucleotide change in a codon of a gene that results in a 1 amino acid change in a protein being made. One more time. Listen to the word change. It's a 1 nucleotide change of in a codon of a gene that results in a 1 amino acid change in the protein being made. All right, now here comes the question. Get ready with your fingers. Get ready, because I'm going to ask you to do a lot of typing on this figure. In pathology, what's the best example of a disease that is due to a missense mutation? The best example of a disease that's a missense mutation disease. That's right. The best example is sickle cell anemia, where we talked about it yesterday. Remember position 6, which is near the N-terminus. There is an A replaced by a T, and as a result, the codon for glutamate shifts for the codon for valine. Glutamate is negatively charged. Valine is hydrophobic. Beta globin doesn't work very well. Hemoglobin doesn't work very well. Red blood cells sickle, and as a result, that give rise to major pathology. All because one little defect in biochemistry. Think about it. One little defect in biochemistry gives rise to major pathology. There are many diseases that are due to missense mutations. PKU, for example, galactosemia, for example, G6PD deficiency, for example. These single nucleotide change, which results in single amino acid change, missense mutations here. All right, now let's continue on. So this is in the second position. Now suppose in the second position, in the normal gene, there's a T here. Suppose the DNA polymerase changed that T not to a C, but to a G. Again, the DNA polymerase makes a mistake. It puts a G where T should be, but the DNA polymerase did not pause and check and correct itself. As a result, the G remains. And now, look at all the other nucleotides, all the other codons are normal, but at this position, notice what we got. At that codon now is TGA. And TGA in DNA means UGA in RNA, and you recognize UGA as the University of Georgia. Without looking in the genetic code, that's the University of Georgia, and you know the University of Georgia is a stop codon. The stop codon normally should be downstream, so now the stop codon is upstream, and this is what's referred to as a nonsense mutation. Here comes the definition. A nonsense mutation is a one nucleotide change which results in a premature stop codon in the gene making the RNA, making the protein. So again, one more time, a nonsense mutation is a one nucleotide change in a gene which results in a premature stop codon in the RNA being made. The premature stop codon should be downstream. Now it's upstream. All the other nucleotides before and codons before the premature stop codons are normal. So now let me ask you a question here. What do you envision the protein to look like in a nonsense mutation? Let me see your answers here. In a nonsense mutation, what do you expect the protein to look like? That's right, everybody. Keep answering. That's right. It would be shorter than normal. It looks small. It would be truncated because it's not getting the amino acids, the later amino acids. So so nonsense mutations result in truncated, shorter proteins. Now, you can imagine that's going to have a major deleterious effect in the cell, the organ, and the body. Here it comes in pathology. How many diseases? If you think about it now in pathology. Dr. Barone will be here a week after next. Again, you're going to love Dr. Barone, his lectures. But in pathology, do you hear a lot about diseases that are nonsense mutation diseases? There aren't that many diseases that are known to be. There are many diseases known to be missense mutation diseases, but there are not that many diseases, just a handful, that are known to be nonsense mutation diseases. Now, why do you think that is? Before I give you an example, why do you think in pathology you don't see very many examples of a disease that's due to a nonsense mutation? Why do you think? That's right. That's right, Tokir. That's right, because and Saga. That's right. It is incompatible with life. Look at this. Look. In missense mutation, it's a one nucleotide change in giving rise to a one amino acid change in the protein being made. And look how devastating that is in sickle cell. In the beta globin gene, you have one amino acid change in beta globin, and it gives rise to devastating sickle cell anemia. But in truncated proteins, it is devastating. It is usually incompatible with life. That's what you need to know. That's why you don't see this very much in pathology, because it's incompatible with life. There it is. There it is. Okay, so so that is the nonsense mutation. But there are a few examples. That are due to nonsense mutation. Does anybody know? Does anybody know in pathology what's an example of a disease that is due to a nonsense mutation? Not that many. Okay, and the best example is one of the hemophilias. And which hemophilia, which protein factor? By the way, in hematology, if they ask you which protein factor is this or that,
If you don't know the answer, what are you going to say in hematology in the blood clotting cascades? If you don't know the answer of the hemophilia, what are you going to say? That's right, factor VIII. That's right. Now, factor VIII could be defective for many other reasons. People have hemophilia because of factor VIII deficiency. Some people have factor VIII deficiency because of missense mutations. But there are few people that have hemophilia due to factor VIII deficiency because of nonsense mutations. The premature stop codon is near the C terminus. So the factor VIII is just missing a few amino acids on the C terminal side, which is near the end of the protein here. So the idea is, but there aren't that many, but in case they ask you what's an example, you say one of the hemophilias. There aren't that many examples. So now let me summarize thus far. High yield. So silent, missense, nonsense mutations, these are what's known as point mutations. Point mutations, listen to the word, are nucleotide change. Change. A point mutation is a single nucleotide change. Why is that so important? Why am I emphasizing the word change? Because there are other mutations that are not due to change. One of them could be due to a deletion. All right, here it is. Now see this nucleotide in the second position. See that TTA codon? Suppose that T in the second position was deleted. It's deleted. And so if it's deleted, the A in the third position now goes to the second position. And the C in the first position of the next codon goes to the third position of the previous codon. So now the C goes in the third position. So in other words, just picture, if you delete one nucleotide, all the nucleotides after that will move up by one position. That's called a frame shift. So one nucleotide one nucleotide deletion results in a frame shift because all the nucleotides, all the codons after that deletion of a single nucleotide are all messed up. And so all the codons are now messed up because the frame shifts. Now if you ask, how did the DNA polymerase delete that nucleotide? Well, what happens there is, again, just picture, in the germline of an individual, when the DNA polymerase is replicating somebody's DNA, when the DNA polymerase is working, replicating some chromosome, and when the DNA polymerase got to some gene, suppose when the DNA polymerase is working on a particular gene to replicate it, what happens is RNA polymerase walks by. So RNA polymerase walks by, and DNA polymerase now looks at the RNA polymerase. Hey, RNA polymerase! Ooh, how you doing? How you doing, RNA polymerase? And then DNA polymerase didn't see that nucleotide. Just didn't see it because it was distracted by something else happening in the nucleus. So DNA polymerase didn't see it like it should have been, so it did. That's why it got deleted. It just happened to miss it. And just tragically, it misses it. So as a result, so it's now deleted, didn't see it when it was working, and then again, all the other nucleotides are normal, but now the frame shifts. Notice now, at the site of the mutation, of the deletion, that new codon is not, instead of being TTA, is now TAC. And TAC in DNA means UAC in RNA, and UAC in RNA in a genetic code means tyrosine. CTA in DNA means CUA in RNA, that means leucine. And there'll be a stop codon, something along that line. So that is, again, that is now where all the nucleotides afterwards causing the codons now, it will be shifting of the codons, giving rise to, you can imagine, the protein being made have normal amino acids, but at the site of the deletion, all the amino acids afterwards are all messed up. And again, in pathology, it's usually lethal because all the amino acids are messed up. But there are a few examples of diseases that are due to frame shift, a one nucleotide deletion. Get ready with your fingers. Get ready with your fingers and try and get your recall from pathology when you have pathology in the second year of medical school. What's an example of a disease in pathology? Best example of a disease in pathology that's due to a frame shift, a frame shift. And the answer is Ricky and Bobby, I need you later. Niu, I need you later. But the answer, Simon, Samanthony, I need you later, not now. The answer is, again, deletion causing a frame shift. The best example is Duchenne's muscular dystrophy. Duchenne's muscular dystrophy. Okay. All right, now in Duchenne's, let me see. What's the name of the protein? What's the name of the protein that's defective in Duchenne's? Name of the protein. Let me see the answer. That's right. It is dystrophin. That's right. Dystrophin is, see, this is my wife. See, there's my wife. She can't. Hey, say hello. They want to say hello to you. What's the matter with you? Okay. What's the matter with you? Okay. All right. Hey, the students are on board, you know. They came on board. They gave me a chance. All right. All right. She spent all my money. She's shopping. You see that big bag of things, you know, all that stuff. She got a big bag. She spent all my money. All right. But in any case, now, what we got over here is, that's right. That's right. Okay. Now, the idea is Duchenne's. It's due to a deletion causing the frame to shift. Now, let me ask you this. In pathology, what's an example of the disease that's very similar to Duchenne's muscular dystrophy, but it's much milder, much milder? Which one is that one? That's right, it's Becker's. Well done, everybody. It's Becker's muscular dystrophy. It's much milder. But now, why is it? You can memorize all you want. You know Duchenne's is severe, MD, and Becker's is mild. But what's the biochemical basis for this? And the reason is simple. Because Becker's muscular dystrophy is an in-frame mutation. It is in the dystrophin gene. It's a missense mutation. It's in frame. There's one nucleotide change, and all the nucleotides, all the codons are normal. That's why it's mild. But in Duchenne's, it's very severe because of the frame shift. Because all the dystrophin protein has many weird amino acids, and that causes dystrophin not to work properly. All right, now question for you is, can you have a frame shift due to a one nucleotide deletion? Can you have I'm looking for the yes or no as an answer. Yes or no. Can you have a frame shift if you have a two nucleotide deletion? Let me see your answers. Two nucleotide deletion. Can you have a frame shift? And the answer is yes. Three nucleotide deletion? Three nucleotide deletion? No. Four nucleotide deletion? Yes. Five nucleotide deletion? Yes. Six nucleotide deletion? No. Seven nucleotide? Yes. So the answer is here, so you can have a frame shift as long as it's not multiples of the deletion, not multiples of three, will cause the frame to shift. Deletions of 1, 2, 4, 5, 7, 8 nucleotides and so on will cause the frame to shift. Okay. All right, now, suppose it is a multiple of 3. It will not cause the frame to shift. All right, here it comes. Get ready with the fingers. Let the fingers do your talking. In pathology, what's the best example of a disease that's due to a 3 nucleotide deletion? Now, Samantha, I think you got it earlier. What's the best example of this? Simna. There we go. Well done. That's right. That's right. That's what I was waiting for you. That's right. And that best example is the all-important cystic fibrosis. Now let me ask you this. What is the name of the protein that's defective giving rise to CF? What's the name of the protein? And the name of the protein, that's right, is CFTR. So it's the chloride channel protein. Now the important feature here is in pathology, 70%, not all cystic fibrosis patients have the same mutation. But 70% of the CF patients do have the same 3 nucleotide deletion. And in pathology, what is that specifically called? Gia and I think Daniel said it earlier. What is the name of the mutation specifically called of the 3 base deletion? That's right. Gia, that's right. Luis, okay. And the answer is, there we go. Now we're talking. And it's called delta F508. Delta means deletion. F means phenylalanine. And the number 508 means at position 508. So there's deletion of the amino acid phenylalanine at position 508. Now let me ask you this. Is the deletion of the phenylalanine in the chloride channel protein, is it near the N terminus or is it near the C terminus of the chloride channel protein? Is it near the N terminus? Is it near the beginning of the protein? Or is it the deletion of the phenylalanine towards the end of the protein? That's right, it's towards the end of the protein because these triple digit numbers, that's telling you that's near the C terminus. It's near the carboxy terminus. It's near the end of the protein. Remember that protein synthesis always begins with amino terminus and you finish with a carboxy terminus. So any amino acid position with single digits, it's near this N terminus, the amino terminus. If you see triple digits, it's near the C terminus. As an example, once again, I hate to bore you with this, but just to drive home the point, I'm trying to drive home the point. In cystic fibrosis, right up above, in sickle cell anemia, the mutation is near at position 6. So in beta globin, there's a glutamate converted to valine at position 6 near the N terminus. But in cystic fibrosis, the deletion of 3 nucleotides is near the C terminus of the protein being made. So again, these numbers are important to try to visualize where the mutation is. All right, now next question. So these are deletions that cause or do not cause a frame shift. What about insertions? If I have a 1 nucleotide insertion, will that cause the frame to shift? Yes or no? 1 nucleotide insertion. Will that cause the frame to shift? Yes, right. And the answer is yes. It causes the insertion will cause the frame to shift. What about a 2 nucleotide insertion? Yes or no? 2 nucleotide insertion. Yes. 3 nucleotide insertion? 3 nucleotide insertion? No. 4 nucleotide? Yes. 5? Yes. 6? No. So the idea is similarly to the deletion. As long as the insertion is not multiples of 3. 1, 2, 4, 5, 7, 8, insertion of nucleotides will cause the frame to shift. Get ready. I forget who answered it earlier. Get ready. Get ready with your fingers. From pathology. From pathology, what's the best example? No, Ahmad, not yet. I need you, no, not yet. From pathology, what is the best example of a disease that's due to a 4 nucleotide insertion that causes the frame to shift? Anybody know? Not Fragile X. I need you later, Katan and Ahmad. Need you later. There we go, Gia. That's right. Ah, Takir, that's right. It is Tay-Sachs disease. Now, Tay-Sachs disease is due to a 4 nucleotide insertion. 4 nucleotide insertion in the gene for hexosaminidase A. And as a result, it messes up the splicing. Now, let me stress something about Tay-Sachs as a disease. You got to know Tay-Sachs. It's one of the most important diseases for boards. We'll talk about it later on in detail, but it's used to illustrate many principles on the boards. It's used to illustrate an example of a disease that's due to an inclusion body disease. Remember, GM2 ganglioside accumulates. It's used to illustrate the disease that's due to an insertion causing the frame to shift. And as a result, it's also used to illustrate the principle of a splicing defect because the frame shifts. It messes up the splicing of the intron, one of the introns for hexosaminidase A. As we'll talk about later on, a couple of days from now, Tay-Sachs, because it's in a religious group, It's used to illustrate the principle of Hardy-Weinberg equilibrium. We'll talk about it a little bit later. So you're going to see Hardy-Weinberg and using Tay-Sachs as an illustrator. You got to know Tay-Sachs for board exams because it illustrates many principles here. Okay, now the last thing here. So as long as the insertion is 1, 2, 4, 5, 7, 8, and so on, it causes the frame to shift. But if it is multiples of 3, then the frame will not shift. Okay, now, Katan, now I need you. Let me see your answer. And Ahmad, what about an example of a disease in which there's 3 nucleotide insertion that does not cause the frame to shift? There we go, finally, at the bottom of the page, that's right. And the best example is Huntington's disease, also Fragile X. Some of you said Fragile X, but Huntington's disease. Let me ask you this, what's the name of the protein that's defective in Huntington's disease? The name of the protein. The name of the protein. That's right, it's called Huntington is the name of the protein. And Huntington is a scaffolding protein to which other proteins bind, and that gives rise to that major disease called Huntington's disease. Now, these 3 nucleotides that insert, that do not cause the frame to shift, they can actually multiply. Instead of having 1 codon insertion, you have 2, 3, 4, 5, and so on. And these are what's referred to. By the way, before we leave this figure, look at the high yield. What do you want me to say? This is high yield. You got to master the basis of the pathologies. And not just the pathologies, the clinical manifestation of the diseases. They might ask you the biochemical basis of the pathologies. What's causing the pathologies? You're looking at such high yield issues here. Now regarding Huntington's disease, these are the trinucleotide repeat expansion, sometimes called a triplet expansion repeat diseases. These are where we got 3 nucleotides that can be amplified or inserted, 3, 6, 9, 12 nucleotides inserted in a gene. And there are 5 diseases. Now, in this paragraph, I'm not going to read it for you, but I'm going to underline just to make sure you know, because they might ask you, which one of the following diseases is due to a 3 nucleotide insertion, multiple copies of the 3 nucleotide insertion that does not cause their frame to shift? And 2 of the 3 that are Huntington and Fragile X. These are the 2 most important of the 5. A third one in this paragraph is myotonic dystrophy, and why I'm showing the paragraph, a 4th one that's buried in the paragraph, so make sure you highlight it because this is the 4th one, is spinal bulbar muscular atrophy, is the 4th example of a trinucleotide repeat expansion disease, and the 5th one is what? Not shown on this figure, not shown on this figure, without looking at Gia's answer here. What's the 5th example? The 5th example, without looking at Gia's answers, that's right, it is, that's right. Well, now we can start typing it up at it, and the answer is, it's Friedreich's ataxia. So 5 different diseases are these trinucleotide repeat expansion diseases that do not cause the frame to shift. Now, some books, like First Aid and so on, show you what is the codon that's being amplified. That's low yield. I doubt they're going to expect you to know what's the codon that's amplified in Friedreich's ataxia. Don't go wasting valuable time. That's low yield item. I cannot guarantee it. I cannot guarantee it, but it's low yield. Just know what are these the 5 diseases. Now we'll talk about later on in genetics that when the codon continues to increase, like in Huntington's disease, the codon is CAG. I doubt they expect you to know that, but CAG is the codon. And notice here, in successive generations, you can have 30 to 60 CAGs, one after another. CAG, 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 one after another in the gene, which makes the messenger RNA, and then the messenger RNA translates the CAG into extra glutamines. So in the Huntington protein, there'll be glutamine, 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 one after another, as you might imagine. The Huntington protein doesn't work very well, giving rise to the tragic disease called Huntington's disease. So that's called anticipation. Now we'll talk more about the word anticipation. Anticipation means in successive generations, from a grandfather to a father, for example, to a son, to the offspring, and so on, the number of repeats increases, and there's an earlier onset of the disease. That's called anticipation. Now, we'll talk more about that later on in genetics. Don't worry about it now. So the 5 diseases that you see here are the high-yield diseases that you must know. All right, okay, so now what's coming up next is look at the high-yield stuff. I'm not wasting your time. Now what's coming up next is the process so in this chapter 4, we talked about the genetic code, and then we talked about the use of the genetic code, how you use it to sequence DNAs and so on, to take the sequence of a coding strand to make it into a protein. We went through that exercise, and we talked about these trinucleotide repeat expansion diseases. Now what's coming up next is the all-important process called translation. But that figure of translation, meaning protein synthesis, the process of translation is 3 figures from now. What we need to do first, before we get to the process of translation, is talk about 2 preliminary figures that set the stage for translation. And both of these preliminary figures are important to understand. Okay, and the first of these is the first of the 2 preliminary figures that set the stage for translation protein synthesis is this. It's called activation of the amino acyl tRNA. Now remember in chapter 3, at the end of chapter 3, we talked about a tRNA, transfer RNA. Remember, I had the 5' end here, it loops around, here's the anticodon at the bottom, and at the 3' end, there is the hydroxyl to which an amino acid will be added. Let me see your answers here. What is the sequence? Again, what is the sequence without looking on the 3' end of all tRNAs have? What's that sequence again? There we go. Well, man, that's good, everybody. You're doing good. That's right, the CCA sequence on the 3' end. So here we have an amino acid here. And then again, translation occurs in the cytoplasm. Replication occurs in the nucleus. Transcription also occurs in the nucleus. Remember, we talked about that earlier. And that transcript can be processed. We talked about that earlier. And then the messenger RNA goes into the cytoplasm. That's what's coming up in this chapter, protein synthesis. So in the cytoplasm, now think about the numbers game here. Think about the numbers, the number of molecules here. It's staggering. What happens is there's a series of enzymes in the cytoplasm. And all these enzymes, for your purpose, just know them as the amino acyl tRNA synthetases. There are a different number of these enzymes, and they use different tRNAs and different amino acids. Now let me ask you this. Now just try, I'm setting this stage to try to understand the concept of what's happening in the cytoplasm. Let me ask you this. You know there's 20 amino acids. We talked about that earlier in the cytoplasm. How many tRNAs are there? Now think about it. Minimally, how many different tRNAs have to be in the cytoplasm? Let me see your answers. Now think. How many tRNAs are there? Not 20. No, Sagra, there we go, Sagra, Tava, Omar, that's right, 61. 61 different tRNAs. Now, how do you know that? How did you get 61? And the reason is because there have to be 61, because you know there's 61 different codons, right? 61 different codons, the genetic code. And that means there are going to be at least 61 different anticodons, meaning 61 different tRNAs. Each tRNA has an anticodon that's specific to a certain codon. So if there's 61 different codons, there got to be 61 different tRNAs and 20 amino acids. Now, continuing on with the numbers game. Again, this is staggering. If there's 61 different tRNAs and 20 amino acids in the cytoplasm, what about the number of the amino acyl tRNA synthetases? There's got to be at least 61 of those enzymes. It means 61 different synthetases. Why? Because each synthetase enzyme has to recognize at the bottom of the tRNA what is the anticodon down there. Is that the anticodon for serine? Well, then the enzyme will put serine on the CCA end at the top of the molecule. And then if this is amino acyl tRNA for glutamate, it recognizes what's the anticodon for glutamate. Well, then it puts glutamate at the CCA end. So again, just picture, you have 61 different enzymes. Each enzyme recognizes which anticodon it's going to look for, and then puts in the proper amino acid at the top of the molecule. 61 different enzymes, 61 different tRNAs, and 20 amino acids. It's amazing what's going on there in the cytoplasm. All right, now don't worry about individual names of the synthetases. Just know them collectively as the amino acyl tRNA synthetases. Now, what do these enzymes do? Now, this is going to bring up a major concept that I want you to understand. Concept, not the detail, the concept. What these enzymes do, to put the amino acid on the CCA end of the tRNA, all these synthetases require energy in the form of ATP. So ATP is consumed down to AMP, and then split off inorganic pyrophosphate. Two phosphates linked together is inorganic pyrophosphate. Inorganic pyrophosphate. And it just so happens that inorganic pyrophosphate is high energy. PPI is a high energy molecule. And also, it's toxic to human cells. So the body knows this. It's toxic. So what the body has is an enzyme you don't have to worry about, low yield. But what the enzyme does, when PPI is generated, it cleaves it right down the middle. It releases the energy and splits off both phosphates as inorganic phosphate. So PPI, inorganic pyrophosphate, is cleaved right down the middle, generating two molecules of inorganic phosphate, liberating all of the energy that was in the PPI. Okay, and then the rest of the energy that's in the ATP is captured in the amino acid linked to the CCA end. And that is a high energy bond. Don't worry about the details, but when the amino acid is linked to the tRNA, it's a high energy molecule. Let me stress one more time. When the amino acid is linked to the tRNA, it is a high energy molecule. Here, it's called amino acyl tRNA, is a high energy molecule. All right, now, you can memorize all you want. You can memorize all you want. But here comes a major issue, is why must the amino acid be activated to make proteins? Why doesn't the body, just hold on right here. Here's Legos. I take it from my grandkids. Legos. See these Legos? Suppose this is valine and this is glutamate. Why doesn't the body take these Legos and join them together and make a peptide bond? It doesn't happen. You cannot take valine, you cannot take glutamate and join them together like Legos to make a peptide bond. It doesn't happen. The amino acids must be in the activated state. And what we're trying to do here is understand why. Matter of fact, before we get to metabolism coming up later next week, before we get to metabolism, the four major classes of macromolecules that precursors must be in the activated state. You already saw that with DNA and RNA. Remember way back on page 19 when we talked about DNA synthesis and RNA synthesis by DNA polymerase and RNA polymerase. Remember, DNA is made up of nucleotide monophosphates, but the precursors to make them were nucleotide triphosphates, the high energy triphosphates. To make RNA, RNA is made up of nucleotide monophosphates, but to make RNA, you need the high energy triphosphates. So to make nucleic acids, the precursors must be in the activated state. Now next week, when we talk about carbohydrates, like glycogen, to make glycogen, to make glycogen, do you take glucose and glucose, join them together like Legos to make glycogen? No. The glucose precursor, the glucose must be in the activated state. Now let me see your answers here. What is the high energy form of glucose? Let me see the answer. What is the high energy form of, not glucose 6-phosphate, Hilal, good try, but glucose 6-phosphate's low energy. What is the high energy form of glucose? And the answer is, it's a molecule called, well done, Andrew, it's called UDP glucose. UDP glucose is the high energy form of glucose. So anything in the body that has glucose in it, like glycogen, like glucose cerebroside, anything that has glucose in it, glucose is donated by the high energy form of it called UDP glucose. Anything in the body right now that has galactose in it, like your blood groups, like gangliosides, the galactose must be in the high energy state. And the high energy form of galactose is something called UDP galactose. Anything in the body right now that has glucuronic acid in it, like hyaluronic acid, like bilirubin, the glucuronic acid must be in the high energy state called UDP glucuronic acid. So the sugars must be in the activated state to biosynthesize complex carbohydrates. What about lipids? Let me see your answer here. To make fatty acids, before we talk about it next week, how do you make fatty acids? Do you take acetate and acetate, join them together like Legos to make a fatty acid? No. The acetate must be in the activated state. What's the activated form of acetate? That's right. Funmi, that's right. It's acetyl-CoA. Marcelo, that's right. Acetyl-CoA. The high energy form of acetate. Hot acetyl-CoA. Next week when we talk about cholesterol, the high energy form of the molecule there is called HMG-CoA. The high energy form of HMG. Now, so again, nucleic acids, carbohydrates, lipids, and now proteins. We're on the topic of proteins. To make proteins, the precursor must be in the activated state. So when we get into the topic of biosynthesis, in the human body, when you make anything, it costs energy. Please keep that in mind. If you want to understand, if you don't want to memorize, but you want to understand big pictures, you got to know that biosynthesis costs energy. As you see right here, to make proteins, to biosynthesize proteins, you need the high energy form of amino acids. Now, on the left side, see these three letters? W, H, Y. Okay, let me see your answers. Why? Because you have to, or do you know why? Why must, now think about it. Why must there precursors to make anything? Why is biosynthesis, before we ever get to metabolism, why is biosynthetic to make glucose and gluconeogenesis, to make fatty acids, to make cholesterol, to make pyrimidines? They must be in the activated state. Why? You need the energy, but why do you need the energy? Why? Why? Because it changes the form. No, I need detail. No, keep trying. If you, I can't read it. Kinetically favorable. Now we're getting somewhere. Okay, now and the reason is simple. The reason is simple. Don't make it hard. A lot of you fear biochemistry. Don't fear it. Try to understand it, and it makes sense. It makes sense. And the answer to that question is it harkens to something you had way back in high school. In high school is where you first talked about the term called entropy. Entropy. In other words, if I take an amino acid that's high energy, another amino acid, and join them together, I'll lose the energy, drive the formation of a covalent bond. And the covalent bond is stable. That's why the precursors are in the activated state, because when I form a covalent bond with sugars or fatty acids or cholesterol, whatever, when I make carbon-carbon bonds and so on, when I join carbon together, I lose energy. You drive the formation of the product. The product is now stable. It's energetically favorable. Please understand what the body is doing. That's what you may, I'm telling you right now, you may hate chemistry. Your body loves chemistry. It appreciates chemistry. It understands chemistry. It lives chemistry because it knows what to do. And so if your body understands it, you should understand it, right? You should understand what the body is trying to do. That precursors must be in the activated state. All right, now what we're going to do is right now it's 5 minutes to the hour. We're going to take another 10-minute break. And then I will be back and talk about the second of the preliminary figure, and that is, what is a peptide bond? After we do that, we'll get into the high yield figure of translation. So let's take a 10-minute break and see you in 10. Okay, everybody, welcome back for hour 3, and we're on the topic of protein synthesis, and we're talking about the activation of the amino acyl tRNA. So again, I want to emphasize that the amino acid must be activated. It's going to cost energy to make peptide bonds and proteins and things. Now, how much energy? Because even though 1 ATP is being consumed here, how much energy ultimately is being used to make 1 peptide bond? And the answer is 2 high energy bonds ultimately will be lost per amino acid. 2. Now, where are the 2 coming from? The high energy bonds. 1 right here, this PPI will be cleaved. You lose energy there. And the energy in the amino acyl tRNA, that also will be lost when we make the peptide bond. So that's why there are 2 high energy bonds per peptide bond showing up here. All right, now, so the next preliminary figure is, what is a peptide bond? What is a peptide bond? And the answer is, what it is the reaction product of an alpha carboxyl group. So this is the alpha carbon. Attached to it is a carboxyl group attached to the alpha carbon. And every amino acid have alpha carboxyl group and alpha amino groups. So here is the alpha amino group of another amino acid like tyrosine. Here is the alpha carboxyl group of methionine. So every amino acid, you got 20 of them. Every single one has alpha carboxyl groups. Every single amino acid has alpha amino groups. And what a peptide bond is the condensation of an alpha carboxyl group of one amino acid and an alpha amino group of another amino acid, forming the peptide bond. And then the energy of the tRNA is lost, driving the formation of the stable bond called the peptide bond, the reaction product of alpha carboxyl group, alpha amino groups of amino acids. How stable is that bond? Well, at dinner tonight, if you had some meat, you had a piece of chicken or a piece of beef or something like that, if you had a hamburger, that food right now is in your intestine. All that meat, all that protein from the meat is now sitting in your intestine. And you cannot break these bonds by themselves. These bonds don't come apart. You have to have enzymes encoded by they're coming from your pancreas. Your pancreatic enzymes like trypsin, chymotrypsin, aminopeptidases, and so on. And what they do is they cleave the peptide bond because they are stable bonds. So proteins are stable, the peptide bonds. And to break the bonds, you need enzymes to break those bonds here. So again, the concept why the amino acid must be activated, you're going to make a stable bond. In the ribosome, the enzyme that carries out this reaction, forming the peptide bond, is called peptidyltransferase. So this is an enzyme. Actually, technically, it's called a ribozyme. It's really technically, it's not a true enzyme. An enzyme is a protein carrying out a reaction. A ribozyme actually is RNA carrying out a catalytic reaction. So one more time. When you talk carrying out a catalytic reaction, you have enzymes, which are proteins. But there are rare exceptions to this, and they're called ribozymes. And ribozyme is it's a piece of RNA that carries out that catalytic reaction. So again, peptidyltransferase technically is a ribozyme, but it's located in the ribosome to make the peptide bond. Okay, everybody. Now we're done with the preliminary figures here, and now this sets the stage for what is translation. So let me just stress before we continue on. Let me stress. The next figure is high yield. The figure after that is high yield. The figure after that is high yield. And the figure after that is high yield. And then we're done with the chapter. Back to back to back to back high yield figures.
I'm not wasting your time. So please pay attention. A lot of high yield stuff coming your way. A lot of integration with farm and path and microbiology and so on. So please pay attention. A lot of immunology coming up. So please pay attention here. Not wasting your time. All right, now what's coming up here is this first of the four back-to-back high yield figures is on page 56, and that is the process of translation. Translation means protein synthesis, right? Replication means DNA synthesis. Transcription means RNA synthesis. Translation means protein synthesis. All right, now the important feature here is that now on this figure, why is it so high yield? 4 disciplines: pathology, microbiology, pharmacology coming up, and of course biochemistry. Biochemistry is everywhere, right? So you know biochemistry is coming up on this figure. 4 disciplines on this figure here. But right at the beginning, I just want to integrate biochemistry with microbiology. So in parentheses, when you see PR, that stands for prokaryotic situation. If in parentheses you see EU, that is the eukaryotic situation. So in the initiation phase of translation, what happens is, suppose this represents messenger RNA. That was chapter 3. You know where the messenger RNA came from, whether prokaryote or eukaryote. That was chapter 3. So here's the messenger RNA from the 5' end because it says so, and here is the 3' end. And here are some codons in there. So this wavy yellow line represents the messenger RNA. Okay, that if this is in eukaryotes, it got processed. Okay, nothing strange. Okay, now in prokaryotes, if you were asked how does the ribosome know to jump on, you say, well, initially in initiation phase, it is the small ribosomal subunit that recognizes the Shine-Dalgarno sequence. Let me see your answers here. What is the name specifically, without looking in the book? What's the name of the small ribosomal subunit in prokaryotes that recognizes the Shine-Dalgarno sequence? Not 40S, good try. It is the 30S subunit. Recognizes the Shine-Dalgarno sequence. In eukaryotes, what is the size of the subunit that recognizes the 7-methyl-GCAP? Now there you go. That's right. That's the 40S subunit. Okay, so once the small subunit binds to the respective parts of the RNA, then there is the initiating tRNA. And in eukaryotes, it's methionine tRNA. And now, by the way, what does this figure, what is this? Now remember, this is where you got to use your imagination. Remember earlier, a couple times earlier tonight, we talked about the tRNA, where at the top of the molecule, that's where the amino acid is attached to the CCA end. At the bottom of the molecule, that was the anticodon. Now use your imagination. Just pretend you're at Disney World or Disneyland. Use your imagination. Just take this tRNA and turn it upside down. Okay? And now we have the anticodon at the top and the amino acid at the bottom. Some people get so bent out of shape. Don't get bogged down here. Just use your imagination. We're just flipping the tRNA upside down. So the anticodon is at the top and the amino acid is at the bottom. So in eukaryotes, it's methionine tRNA. In prokaryotes, it's F-met tRNA. Now the F stands for formyl. It's next to a carboxyl group. Not a big deal for the boards. So, but the idea, it's F-met tRNA. It's just an extra carboxyl group on methionine is F-met on the tRNA. Now, so the initiating tRNA will then come in, and then the large subunit in prokaryotes and eukaryotes also come in, and to do all this condensation, to bring it all together, IFs, called initiation factors, low yield. Low yield initiation factors. These are proteins that bring everything together. And by doing so, we have now assembled the entire ribosome with the messenger RNA. So now notice here in the ribosome, here again the yellow part represents the messenger RNA, and in the ribosome there are 2 compartments. There's one compartment called the P site, which stands for the peptide site, and there's an A site, which stands for the amino acid site. In the P site, that is where the codon AUG, that's the initiating codon, base pairs with the methionine tRNA. So the methionine tRNA has an anticodon that base pairs with the AUG codon in the P site. In the A site, there's another codon. In this example, it's CUG. Outside, there's another codon AAA. In this example, the rest of the messenger RNA not even shown here. Just use your imagination. This is all during the initiation phase. Please keep that in mind. Pharmacology is going to be crucial in a little bit. I'm not trying to bore you with this information. It's critical for understanding integration. So this is now assembling the ribosome on the messenger RNA. So then what happens? What happens is all 61 tRNAs, every single one, leucine tRNA, lysine tRNA, all 61 different tRNAs with the different anticodons come into the A site. One at a time, they're coming into the A site. One at a time, they come into the A site. Look in the base pair with the CUG codon. And the only one that can do that in base pair is the leucine tRNA. So leucine tRNA has the anticodon that base pairs with a CUG codon. To bring the aminoacyl tRNA into the A site, it requires GTP and these EFs. Now, EF stands for elongation factor, in contrast to IFs, which are initiation factors, low yield in both prokaryotes and in eukaryotes. Now, if you see the little E, that's referring to the eukaryotic situation. If you don't see the little E, it's prokaryote. But these elongation factors, low yield stuff. All right, let's proceed. So then what happens is in the ribosome somewhere is that peptidyltransferase. And what does the peptidyltransferase do? What it does, it condenses one amino acid with the other one by forming the peptide bond. It condenses an alpha carboxyl group with an alpha amino group and to form the peptide bond. So now we have the peptide bond being formed in the A site. And then what happens? And then what happens is the ribosome will now move by one codon position. It's called translocation. The ribosome it will now move by one codon position. So now notice here, when the ribosome shifts by one codon position, then the AUG codon goes outside, and in the P site comes the codon CUG with the anticodon with the peptide linked to the tRNA. And in the A site comes another codon. In this example, it's AAA. So again, don't get bogged down. Just try to picture what's going on. The ribosome now shifts after you make the peptide bonds here. Now, to do the shifting called translocation, it requires GTP and elongation factors as well. And importantly, get ready with a highlighter. There is a major elongation factor called Oh, by the way, if you were asked, again, these two high energy bonds, again, that was the previous page, making the peptide bond. But to do the translocation, it requires a major elongation factor called elongation factor 2. Now, why is that so important? That's going to be discussed in just a few minutes. But right now, we're doing the straightforward biochemistry and microbiology integration here. Okay, so again, so far here. So again, so these elongation factors here are not important, but in the eukaryotes, there's a major one called elongation factor 2, critical. Not elongation factor 1, elongation factor 2. All right, let me ask you a question here. How many high energy bonds total was used to make one peptide bond? You see your answer. How many high energy bonds was used to make one peptide bond? Not two. You're thinking two. And the answer is 4 high energy bonds in the process of elongation. Now, where do the 4 come from? Now, watch. Watch, where does the 4 come from? Notice here, to bring the amino acyl tRNA into the A site, GTP is needed there, high energy. 2 high energy bonds for the activation of the amino acid from the previous page, and then 1 GTP to do the translocation. So 1, 2, 1 high energy bonds were used per peptide bond. So what I'm driving at is protein synthesis is expensive. It's expensive. To make any protein, every peptide bond is expensive. It costs 4 high energy bonds. Now I'm going to ask you a question here. And a lot of you are going to get it wrong because you're not thinking. You're trying to memorize here. Okay. So, and by the way, number crunching for board exams is low yield. I doubt they're going to ask you how many ATPs you get in glycolysis, blah, blah, blah. I doubt that. In your days, that was high yield. But I doubt they're going to do that in recent years. I cannot guarantee they won't. But nevertheless, suppose they say to you, a lot of you are going to get this wrong. Suppose they say to you, a protein has 100 amino acids. 100 amino acids in a particular protein. How many high energy bonds was used to make it in the process of elongation phase? Again, one more time. 100 amino acid protein. How many high energy bonds were used in the process of elongation? Let me see your answers. Give everybody a chance. So I see 400, I see 200, I see 40, I see... There we go. There we go. There, now I see the answers. Tariq and Haik. If I pronounce your name, I apologize. Haizi. But you're right. It's 396. That's right. 396. Omar. That's right. 396. Not 400. Most people think, well, 100 amino acids. That's times 100 times 4 is 400. No. How many peptide bonds are in a protein that has 100 amino acids? And it's 99. 99 peptide bonds times 4 is 396. 396. It's not hard. It's not tricky. I know a lot of you think, oh, that's tricky. It's not tricky. It's a matter of thinking. Thinking. If I said to you a protein has 10 amino acids, how many peptide bonds are in it? It's 9 peptide bonds. So 9 times 4 is 36. Not hard, not hard. It's just a matter of thinking. See, there it is. Just think. Use your mind. Okay, now let's continue on. Continue on with the basic biochemistry and so on, and microbiology. So then what happens? All 61 tRNAs come into the A site. Look in the base pair with AAA, and the only one that can do that is lysine tRNA, peptide bond formation, translocation. Another amino acyl tRNA comes into the A site, peptide bond formation, translocation. Another amino acyl tRNA comes into the A site, peptide bond formation, translocation. So as the ribosome is moving on the messenger RNA, the protein starting with amino terminus is getting bigger and bigger and bigger and bigger until somewhere in the A site comes the Universidad Autónoma de Guadalajara, or the University of Georgia, or the University of Alaska at Anchorage. One of the 3 stop codons comes into the A site, and then as a result, protein synthesis terminates. And then the protein then comes off the ribosome and folds and so on. We'll talk more about protein folding on the next figure. So there it is. There is the straightforward issue of translation. What you need to know and what you don't need to know. There's a lot of stuff here. I could be boring you silly with some useless information. Don't worry about the low yield items. Okay, but now that's the basic stuff of translation. Now that's 2 disciplines so far, biochemistry and microbe. Microbes do basically the same thing, except initiating tRNA is different, and then the Shine-Dalgarno in microbes. 30th subunit, again, that's the size of the subunit different. All right, but now here comes the pharmacology. Because we know the issues of microbiology. In pharmacology, there's a lot. There are many antimicrobials that target page 56. Let's go through a few of them. At the top of the figure, now again, if you have the answers in front of you in your book because you looked at the videos or you looked in first aid, don't answer. I'm trying to get other people to think, try to recall from pharmacology, try to recall from microbiology one by one. So if you have the answer in front of you in your book, don't answer. I appreciate it if you don't answer. Here it comes. From microbiology and from pharmacology, what is the name of that class of antimicrobials that takes advantage of your knowledge that in microbes the 30S subunit recognizes the Shine-Dalgarno sequence, and that drug inhibits the binding of 30S to the Shine-Dalgarno sequence? What's the name of that drug? Not tetracycline. It is, that's right, the first one was, it's the aminoglycosides, like streptomycin and gentamicin. They inhibit the recognition of 30S with the Shine-Dalgarno sequence. Because we don't have 30S, because we don't have Shine-Dalgarno, we have the 7-methyl-GCAP. That's why the aminoglycosides have no effect on me and you. Now another item in pharmacology, and what's the name? And this one might be a little bit harder. What's the name of that another antimicrobial that also inhibits initiation phase? And what that drug does, it binds to the large subunit in microbes and prevents the assembly of the ribosome on the messenger RNA. What's the name of that drug? The key here was the word initiation phase. What's the name of it? Not chloramphenicol. Good try. At least you're trying, not macrolides. You're trying. Even though you're giving the wrong answer, you're trying. You'll never forget the right answer. It's a very expensive drug. It's a chemical, not a, it doesn't come from a fungus like penicillin, but it's a very expensive drug. It's a drug that's used as a last resort to treat MRSA, vancomycin-resistant microbes, things like that. It's a last resort, but it's very expensive. And there we go. Now people are getting the answer, and the answer is it's linozolid. And again, the key there is it binds the 50S subunit, because the words they may use on boards. It binds the 50S subunit and prevents the initiation phase. I cannot emphasize enough the words initiation phase of translation in microbes here. All right, now continuing on in microbiology and pharmacology. In pharmacology, what's the name of that antimicrobial that prevents the amino acyl tRNA from binding into the ACE site? What's the name of that antimicrobial? Now, somebody mentioned that earlier. I saw it earlier. That's right. Now we go. That's tetracycline. Tetracycline. Now, some books like First Aid, what they say is that tetracycline binds to the 30S subunit. That's a matter of semantics. Here, notice that the codon-anticodon base pairing is occurring in the 30S region of the ribosome. What tetracycline does, it disrupts the codon-anticodon base pairing, and therefore microbes cannot make proteins, therefore they'll be eradicated. All right, now next one. What's the name of that drug that inhibits the peptidyl transferase? Well-known antimicrobial inhibits the peptidyl transferase. Okay, and if I said to you, if I use the words gray baby syndrome, everybody will now get it correct. There we go. Little long word there. That's right. That is chloramphenicol. Inhibits the peptidyl transferase. Now, in microbiology, pharmacology, biochemistry, what's the name of that class of drugs that binds to the 50S subunit, but it prevents the translocation of the ribosome in the process of elongation? What's the name of that one? That's right. That's the macrolides. The macrolides, like erythromycin and so on, they bind to the 50S and prevent the process of translocation. Now, there's a related drug, but it's not a macrolide, but it's a related drug, and that's clindamycin. And what clindamycin does, it behaves just like a macrolide. It binds to the 50S and prevents the process of translocation. So again, look at the high yield. What do you want me to say? This is high yield stuff. So again, you must know what is the target and what process is being inhibited. So if they use the word of drug inhibits, binds the 50S subunit, pay attention. Are they talking about the initiation phase or the elongation phase? Because these drugs work differently. So tetracycline, what it does, it prevents the amino acyl tRNA from getting into the A site. Look where it's located. It's right here. It's preventing the amino acyl tRNA from forming the codon-anticodon base pairing in the A site. Therefore, proteins will not be made in microbes. Now again, before I leave this, I cannot emphasize enough. You got to get those antimicrobials straightened out. Yesterday we spent a lot of time talking about the fluoroquinolones that inhibit the process of replication. Yesterday we talked about rifampin that inhibits the process of transcription. Here we're talking about drugs that inhibit the process of translation. You got to get these drugs straightened out. What they target, where they target, what process is being inhibited, and blah, blah, blah. There are other antimicrobials out there we'll talk about later on. They can inhibit DNA synthesis by inhibiting the formation of the DNA precursors and so on. Drugs like Bactrim, sulfa drugs, but they prevent DNA. They prevent DNA synthesis for other reasons, not like fluoroquinolones. The Bactrim drug, it works by preventing DNA precursors from being made. You must get these antimicrobials straightened out. Out of 322 questions on boards, at least one of them, you would imagine, is going to be an antimicrobial, right? The sooner the better. Don't go memorizing, just picture, or just practice, just verbalize the answer. Which drugs target this? It's not hard. It's not hard. Just, you want to know this material. You want to be positive. You want to know this material. It helps your career. All right, now what's coming up next is, where's the pathology at? Well, it's coming up, and there's more microbiology. All right, now this issue of Pseudomonas and diphtheria toxin. That's what's in the pathology of diphtheria and so on. Okay, now what's coming up next is we got to do some biochemistry explanation. As you know from Dr. Muscatello's lectures in microbiology, that Pseudomonas and diphtheria toxin can kill me and you. And if they ask you on board exams, what's the mechanism of killing? You answer by saying by the process of ADP ribosylation of elongation factor 2. Those are just words. What does that mean? ADP ribosylation of elongation factor 2. You can memorize those words. You can look for mnemonics and this. What does it mean? ADP ribosylation of elongation factor 2. All right, now, and because ADP ribosylates elongation factor 2, it doesn't work, and you can't make proteins, and you'll die as a human being. All right, but now we're going to do explain. What does it mean? And it's not hard. Believe me, trust me, it's not hard understanding what it means. Many times I ask medical students, what does that mean, ADP ribosylation? And so many students will say, I don't know. And I said, well, don't you ever ask your instructor? And the students will say, hey, ours is not the question why. Ours is but to do or die. If you never knew what it means, we're going to find out what it means, because it's not hard. And you remember it. You remember it because you see it. All right, now before we talk about what does it mean, ADP ribosylation, let me ask you another question. Here it comes. Get ready. Get ready. Get ready with your fingers to type in the response. And this is crucial. You must understand this next part. What class of protein is elongation factor 2? What class of protein is it? Is it a receptor class? Is it a kinase class? Is it a phosphatase class? What class of protein is elongation factor 2? To make sense out of all this. Not a kinase, not a phosphatase, not a receptor. No, 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 no. And the answer is, it's a G protein. Now, this is the first of many times over the next 2 weeks we're going to be talking about G proteins. And right at the beginning, let's talk about and review what does that mean, G protein. You know, you saw that earlier in physiology from Dr. Wilson's lectures. In physiology with endocrinology, all these G proteins, GS, GI, GQ, and so on. What does G protein mean? And the answer is simple. Any G protein, when you hear the word G protein, it means it binds GTP in order to work. As simple as I can state. Any G protein is it binds GTP in order to work. Now, what about elongation factor 2? Notice right above it, GTP. To make the ribosome move on the messenger RNA, it requires elongation factor 2 that binds GTP in order to make the ribosome move. So elongation factor 2 is a G protein. GS, stimulatory G protein, we'll talk about several days from now, also is a G protein. GI, GQ are G proteins. Why? They bind GTP in order to work. All right, so that establishes what class of protein is elongation factor 2. All right, now the question is, what does the process of ADP ribosylation mean? Now remember the huge integration with microbiology. Because from Dr. Muscatello's lectures, remember there are 3 other toxins: cholera, E. coli, pertussis toxin, and 2 more right now: Pseudomonas and diphtheria. 5 different toxins. All of them work by ADP ribosylation of a target G protein. The target G protein for Pseudomonas and diphtheria is elongation factor 2. The target G protein for cholera is GS, for example. The target G protein for pertussis toxin is GI. We'll talk about that next week. Don't worry about it right now. But what I'm getting at here is there's 5 different toxins, all work by ADP ribosylation. All right, now to explain the biochemistry, it's very simple. What happens is, again, just listen and pay attention. It'll make sense if you think about it. What happens is all 5 toxins, 2 of them right now, the other 3 later on, all 5 toxins actually are enzymes, and they're secreted by the respective pathogenic organisms. And when these any of these enzymes come into your cell, because they are enzymes, they are looking for a substrate. What is the substrate that all 5 of these toxins are looking for? In other words, let me go to the whiteboard here. Suppose this wavy line represents the target G protein. This could be elongation factor 2, the target G protein for diphtheria toxin, or this could be GS, the target G protein for cholera toxin. But when these toxins come into the cell, what is the substrate they are looking for? What are they looking for? What substrate? Not hard. What hard? What are they looking for? And we see the answer. Not GTP. Good try, Lewis. You would think not proteins. The protein is the target. What substrate are they looking for? Okay, here comes the answer. And it'll start to make sense shortly. The substrate they're looking for is a substrate called NAD. NAD. I got a lot to say about it next week. NAD, all this anaerobic situations that go on, lactic acidemia, and that's all next week. But the idea of NAD, they're looking for NAD. Now I'm going to show you the structure of NAD. Not that you need to know it for the boards, but if you see the structure of NAD, you'll start to picture what's happening, and it makes sense. Okay, now here it is. You don't have to write it down. Don't write it down, the structure of NAD. But here it comes. This is the structure of NAD. And NAD comes from the vitamin called niacin. In the yellow is niacin. Now we'll talk about niacin, the vitamin, next week. But when you eat niacin, you make NAD. Now look at the rest of the molecule. Look what the niacin is linked to. Look at the bottom of the molecule. Notice a base, a sugar, two phosphates. Ah, a nucleotide. Base, sugar, phosphate is a nucleotide. Now, which nucleotide is it? So you look at the base and you see two rings. You say, it's got to be a purine. And if it's a purine, it's either adenine or is it guanine. And how do you know, what is it? Is it adenine or is it guanine? That's right, it is adenine. Why? Because at the 12 o'clock position, there is an amino group. So that's adenine plus ribose, the two phosphates. That's ADP. That's ADP. Look how simple that was to figure it out. Look how simple it was to figure this out. There's no magic because you're using your mind. You're using your mind. You're beautiful how the mind works. So this bottom of the molecule is ADP. But look what the ADP is linked to. Between the niacin and the ADP is ribose. So it's ADP ribose linked to the niacin. ADP ribose linked to the niacin is NAD. So what happens is, that's in the cytoplasm. What happens is when a toxin like diphtheria toxin comes into the cell, because it's an enzyme, what it's going to do is it looks for NAD and splits off the niacin. It cuts this bond right here. It just cuts this bond right there and splits off the niacin. That's not the problem. The problem is the rest of the molecule. And what happens is the rest of the molecule is ADP ribose. And the toxin takes the ADP ribose, the rest of the molecule, and puts it on in covalent linkage onto a target G protein. And if this is diphtheria toxin, target G protein is elongation factor 2. And that's now called ADP ribosylation of the target G protein. There it is. It's not hard. Look how simple this was to figure it out. It's not hard. And then what's going to happen in elongation factor 2? When it's ADP ribosylated, it doesn't work. And if it doesn't work, you don't make proteins. There it is. It's not hard. Look at that. Notice how a little bit of biochemistry, a little bit of information goes a long way. It explains things. It makes sense out of things. So when you hear the words coming up next week, with cholera toxin, pertussis toxin, ADP ribosylation of GS or GI, it's the same principle. Same principle. It's just the target G protein is much different. There it is, okay? So one more time. So all 5 toxins, they look for NAD in the cell, and they split off the niacin. The rest of the molecule is ADP ribose, and it's put onto covalent linkage onto a target G protein. And what is the target G protein? It depends which toxin we're talking about. Pseudomonas and diphtheria target elongation factor 2. Put ADP ribose on the elongation factor 2, and it doesn't work. Next week, cholera toxin, ADP ribosylates GS, and then affects that G protein. Pertussis toxin, ADP ribosylates GI, and affects that G protein. So again, the amazing thing. It's not hard to understand. Okay, so again, this is now the first of the back-to-back high yield figure. Notice how high yield this was. Again, as long as you're paying attention, you know what you need to know. Don't go worried about the gory details of translation, all these initiation factors. We covered the high yield topics. There is no reason to read about it unless you want to, because we covered the high yield topics here. Now what's coming up next is another high yield topic, and that's the issue of proteasomes and ubiquitin. Now for a long time, this was not high yield. But in recent years, last 3 or 4 or 5 years, I've been hearing a lot about proteasomes and ubiquitin. And so now these are elevated to high yield status. You must know the issue of proteasomes and ubiquitin because it integrates with many other things here. Now, before we do the integration, let's get into what's this all about. What this is about is simple. What happens is proteins will misfold. And when a protein misfolds, for whatever reason we'll talk about in a few minutes, but proteins misfold. And when a protein misfolds, it's like a piece of junk, a piece of garbage. And the cell wants to be a nice, tidy cell. Remember, cells are in a community of other cells. It's in a neighborhood of cells. And all the cells, they don't want to be the bad player. All the cells want to be nice and tidy, nice and neat. Okay. And so when a piece of protein misfolds, it's a piece of junk, piece of trash, and the body wants to dispose of that piece of trash. It wants to be nice and tidy. It wants to dispose of that trash. So how does that happen? Okay. So in other words, for example, suppose I went to McDonald's and I went to McDonald's and got a Big Mac. And the Big Mac comes in a wrapper. So I come home, I take the Big Mac, I start eating the hamburger, and I take the wrapper and I wrap it up and I throw it on the ground. Within microseconds, my wife will come out and yell at me and grab me by the earlobe. See this earlobe here? I guess that's why we have earlobes, right? Because it's stretchy. So my wife will grab me by the earlobe and drag me to that piece of trash and say, pick up that piece of trash and throw it into the garbage can. Yeah, blah, blah, blah. It's the same thing, right? So grab me by the earlobe. That's the same thing that happens in your cells. Same thing that happens in your cells. A piece of trash is not permitted to accumulate because a cell wants to be nice and tidy. So how do you take a piece of trash, a misfolded protein, and throw it into the trash can? Okay, so what happens here is there is an enzyme low yield. I've not heard about it being on boards, so I would not be concerned about it yet until a student tells me they saw it on the boards. I've never heard about the enzyme. So don't worry about it, low yield. But what the enzyme does, it uses ATP. And what happens is there is a small peptide called ubiquitin. Don't worry about how big it is, but it's a small peptide. And what happens is this enzyme takes multiple ubiquitins, it's a peptide, and puts on the ubiquitins onto the misfolded protein. So now we form polyubiquitinated proteins. So multiple ubiquitins are put onto the misfolded protein. And now that's like a signal to say, oh, okay, take that piece of trash that has ubiquitin on it and throw it into the garbage can. And what is the garbage can? And here it is, in the cytoplasm. There is a particle called the proteasome. It looks like a garbage can. And it looks like a big cylinder. It looks like a garbage can. And the misfolded protein that's polyubiquitinated is thrown into the garbage can. And when it throws it into the garbage can, called proteasomes, the ubiquitin is cleaved off and salvaged and reused over and over again. So one more time. When a protein misfolds, it's a piece of trash. So then there is an enzyme that uses ATP to put multiple ubiquitins onto the misfolded protein, forming polyubiquitinated protein. And that's like a signal to tell the cell, throw that piece of trash into the garbage can, called a proteasome. And a proteasome is a cylinder. It looks like a cylinder, like a garbage can. And so the misfolded protein is thrown into the garbage can. The ubiquitins are cleaved off and reused over and over. Now in the cylinder, in the garbage can called the proteasomes, in the inside are proteolytic enzymes. There are proteases that cleave the misfolded protein into peptide fragments, which can further be broken down to amino acids here. But there it is. That's how, again, this proteasome is located in the cytoplasm. And the idea is that's how these misfolded proteins are degraded in the proteasome. Here. Okay, now let me ask you this. Don't look in your book in case it's in your book. Don't look in the book here or in first aid or some other book. How do you just had Dr. Muscatello in immunology. How do you think this integrates, this biochemistry of the proteasomes and ubiquitin? How do you think this integrates with immunology? Let me see your answer here. How does this integrate with immunology? That's right, Leonardo. That's right. That's right. Okay, how does it integrate with immunology? Because in immunology, when Dr. Muscatello was here last week, the idea is when a phagocyte takes up a microbe, and the microbe is killed by the respiratory burst, and now we have a dead microbe in the cell. And what happens is the proteins from the dead microbe are then processed. These microbial proteins misfold. They are then thrown into the garbage can called the proteasome of the phagocytic cell. And then these peptide fragments are then presented on the cell surface. Antigen presenting cells, APC, antigen presenting cells. So these peptides are now put onto the surface. And as Dr. Muscatello discussed then, that's how you get the immune response. You get antibodies being generated because of the antigen presenting cells. It just makes sense. This is the biochemistry behind the immunology. All right, now another item here, then we'll take the break. So then, now coming back to the normal situation here, is how does a protein misfold? Well, what happens there is, or whatever, when do proteins misfold? It depends what protein we're talking about. Every protein has a half-life. Some proteins, like half-life meaning there'll be a time that a protein will age and the protein dies. I'm going to age and I'm going to die. Same thing with proteins in your cells. Every protein ages and dies. It has to be turned over. That's called the half-life. Now, some proteins have a very long half-life. For example, collagen has a half-life over a month. But other proteins, for example, their key regulated enzyme of metabolic pathways, those enzymes generally have a very fast half-life. For example, HMG-CoA reductase, the top regulated enzyme of cholesterol metabolism, has a half-life of 2 hours. So regulated enzymes have a very short half-life. But every enzyme, every protein will turn over. It depends what protein we're talking about. It just simply, it misfolds. For example, a regulated enzyme, it's working, catalyzing reactions and so on. Eventually, the enzyme says, enough is enough. I'm tired of working, and it misfolds. And then it does not accumulate. It has to be thrown into the garbage can. But where else can we generate misfolded protein? Is new proteins when they're being made on the ribosome. Some proteins start folding and some proteins misfold while they're being made, but they don't accumulate. So as a result, any protein that misfolds while it's being made is thrown into the garbage can, as well as old proteins. Every protein will age and has to be thrown into the garbage can and destroyed. Now, to finish off before the break, there are some proteins that are so complex that they need help folding properly. They cannot fold by themselves, and therefore they need a helper protein to fold properly. And these helper proteins are called chaperones. So chaperones are proteins that help other proteins fold properly. Now, some of these chaperones are called heat shock proteins, HSP70s. I wouldn't be too concerned about that. But just know the concept. A chaperone is a protein that helps another protein fold properly because the protein might be too complex. But an example of a protein that needs a chaperone to fold properly is alpha-1 antitrypsin. Alpha-1 antitrypsin is a protein that's made by the liver only and needs a chaperone in the ER. We'll talk about the ER secretion after the break. We'll talk about secretion after the break. But the idea is alpha-1 antitrypsin needs a chaperone to help the alpha-1 antitrypsin fold properly in the ER network here. Okay, so now let's take a 10-minute break, and then we'll come back for the last hour. So I'm going to go off camera and see you in 10. Hello, everybody. Welcome back for the last hour of biochemistry here, and two more figures to go, and then we'll finish chapter 4. After we finish chapter 4, if George can get ready with the selection pod, I have 2 questions that based on content we've covered at the end of chapter 4. So we'll get to it in about 15-20 minutes, something like that, maybe a little bit longer. But the idea is there'll be 2 questions that are based on stuff we've talked about, and we'll have a selection pod give you a chance on your own. How well do you know the material to figure out the answer? Okay, so anyway, so now that 2 more figures to talk about. So, and the first, and both of them are high yield. And the next figure to talk about is, looks complex, but that is how do secreted membrane bound or lysosomal enzymes, how do they know where to go? You know some proteins like the blood clotting factors are secreted, but how do they know to be secreted? Why don't they sit in the cytoplasm? How do proteins that go into lysosomes like proteases and glycosidases, how do they know to go into the lysosomes? How do proteins like the sodium pump know to go onto the plasma membrane? It's easy to say they do, but what's the biochemistry behind that? That's what this figure, page 60, is what's designed to explain. The biochemistry of how these things happen. Here, don't go memorizing. Just try to understand what goes on. All right, now on the left side here, we got a cell. And in the middle of the cell, in the purple, represents the nucleus. Now, remember, in the nucleus, you have 46 chromosomes. And these 46 chromosomes are all mixed together. You know, a lot of times, a lot of students think that the 46 chromosomes are lined up in some sort of karyotype. Nice and neat, you know, chromosome 1, 2 of them. And no, they're all mixed together. They're all waving, you know, this kind of, they're all mixed together. But the important feature is in the nucleus. That's where the enzyme RNA polymerase is located. And when the signal is given to tell the RNA polymerase, hey, RNA polymerase, go into the chromosomes and find a particular gene and turn it on. So sure enough, RNA polymerase will bind to the promoter for the gene, and then when it binds to the promoter, then the RNA is then transcribed from the gene, and then the RNA is in the nucleus is capped, spliced, and tailed. Capped with the 7-methyl-G cap, spliced with the spliceosome, and tailed with a poly-A tail. This is all going on in the nucleus. Where the RNA, one more time, where the RNA is capped, spliced, and tailed. All right, all going on in the nucleus. And then after all that happens, then the messenger RNA leaves the nucleus and comes out into the cytoplasm. And when it comes out into the cytoplasm, that's where the ribosomes are located. And the ribosomes now will jump onto the 7-methyl-G cap. The 40S subunit binds to the 7-methyl-G cap, and then the initiating tRNA comes in, methionine tRNA. The large subunit comes in, the 60S, bringing them all together to form the ribosome. So that's all going on in the cytoplasm. And so now here is, now if we were to magnify a portion of the cytoplasm, here is now the messenger RNA shown in the blue. So that's the messenger RNA for a particular protein. In the red represents the ribosome, and in the yellow that represents a protein being made off the ribosome. And we always start with the N-terminus. Now notice what I just stated here on page 60. All the stuff that I talked about, nothing new. It was all covered in the previous figures and chapters and so on. There was absolutely nothing new that I mentioned. Now, if you're thinking, oh man, that was new to me, you weren't paying attention when we talked about it earlier. So again, nothing new on what I just described. Now, what's coming up next is new material. Now, any protein that's destined to be secreted, membrane bound, or lysosomal, what happens is on the N-terminal side of the newly synthesized protein being made, there's going to be a stretch. Let me highlight it in the green. There's going to be a stretch of about 10 to 15 hydrophobic amino acids. Amino acids like alanine, valine, isoleucine. So again, there's going to be a stretch of hydrophobic amino acids. And it's sometimes called the N-terminal, because it's on the N-terminus, hydrophobic, because it is, signal sequence. This is now a signal to be recognized by a particle called the SRP, which stands for signal recognition particle. This is a particle in the cytoplasm. Now, again, don't get this confused with the spliceosome. The spliceosome also is a particle. It's in the nucleus to do splicing. The SRP is in the cytoplasm. And what the SRP is, it recognizes, it binds to that N-terminal hydrophobic signal sequence of the protein destined to be secreted, membrane bound, or lysosomal. So what happens is the SRP binds to that stretch of hydrophobic amino acids, and then what happens is the SRP attaches the entire complex on the outer leaflet of the ER membrane, the endoplasmic reticulum membrane. Remember all the cell biology? So now we have the ribosome with the messenger RNA facing the cytoplasm attached to the ER membrane. And then what happens? The SRP opens up a little channel to which the peptide is now threaded through the channel, and the peptide now comes into the lumen of the ER. It's like an eye of a needle. So a piece of thread is being threaded through the eye of the needle because a little channel is opened up, and the protein now goes to the lumen of the ER. While the protein is being made, co-translational, while the protein is being made. Okay? So again, so one more time, you must understand this. Not hard, I'm making it simple. Don't get bogged down on details. Understand concepts here. But the idea is any protein that's destined to be secreted, membrane bound, or lysosomal must have an N-terminal hydrophobic signal sequence. And that's a stretch of hydrophobic amino acids, 10 to 15 hydrophobic amino acids, right in a row on the N-terminal side. Why that's important? Because it's recognized by the signal recognition particle, binds to it, and then attaches the ribosome and the messenger RNA on the surface, the outer side of the ER membrane. And the peptide is now threaded through the membrane into the lumen of the ER. In cell biology and in histology, when the ribosome is attached to the ER membrane, you might see it called the RER, rough endoplasmic reticulum, where the ribosome is attached to the membrane. When the ribosomes are not attached, it's called the smooth ER. But again, when the ribosomes are attached, it's called the RER, rough endoplasmic reticulum. All right, let's continue on. Again, this is all while the protein's being made. By the way, let me ask you this. Just think what I just mentioned, the new material. Suppose they say to you, you know how they can ask you about this? Suppose they say to you, and there is a mutation in codon 2 of prothrombin. What would that do? Again, one more time. The gene for prothrombin has a mutation at position 2 of the codons. What would that do to prothrombin? Let me see your answers here. Now think. So prothrombin has a mutation at position 2. What would that do to the prothrombin? Misfolded, that's true, but even more important, Non-functional? No. Well, it's true too. That's true. And the answer is it impairs secretion. It would never get out into the blood. Never. It would impair the secretion because if there's a mutation in the first 10 or 15 codons, then you don't get the N-terminal hydrophobic signal sequence. Maybe it makes it into a polar amino acid depending on the mutation. And SRP will not recognize it. It will not attach it to the ER membrane, and therefore it stays in the cytoplasm. It would never be secreted. That's how they can ask this on board exams. Please think globally, think bigger picture. One more time. If there's a mutation at the first few codons, a missense mutation such that it converts the hydrophobic amino acid into a polar amino acid, something like that, then the SRP doesn't recognize it, doesn't bind to it, will not attach it to the ER, never gets in the lumen of the ER, and never be secreted. Impairs secretion. There it is. It's not hard, as long as you're thinking, oh yeah, it makes sense, right? It makes sense. All right, let's continue. Now let's continue. So then, coming back to the normal situation. So the protein now, while it's being made, it's getting bigger and bigger and bigger. The ribosome is moving along the messenger RNA. And on the protein is getting bigger, bigger, and bigger. And again, it's now in the lumen of the ER here. While it's being made, a process called N-glycosylation can take place. N-glycosylation, what that means is there are enzymes you don't have to know. Don't worry about them. But there are enzymes that now will put sugars onto the nitrogen of a particular amino acid. Does anybody know what amino acid we're talking about? Which amino acid? Which amino acid? Not arginine. Good try, but it's not arginine. Good try. Arginine does have nitrogen. Not methionine. Good try. Not histidine. Good try too. And there we go. Dinesh. That's right. And Ifosa. That's right. It is asparagine. Asparagine. So in other words, proteins now will have sugar chains attached to the nitrogen of particular asparagines. So in this example, here's a protein being made. These little dots that you see right here represents sugars attached to asparagine. Right here is another asparagine to which there's sugar chains attached while the protein is being made. So now because there's sugars on the protein is now called a glycoprotein. Glycoprotein, there's sugar on the protein. And then the protein is finished being made. Now again, I want to stress, N-glycosylation is co-translational. Listen to the words, co-translational. While the protein's being made, sugar chains are added. Okay, co-translational. And depends on the protein. Some proteins may have one sugar chain, other proteins may have two sugar chains, and so on. And then the protein enters the Golgi apparatus and then can be secreted. Now let me ask you this. Don't worry about the details, what kind of sugars, this kind of stuff. I doubt they're going to ask that. But let me ask you this. The proteins that are in your bloodstream right now, the proteins that are on your plasma membrane of all your cells, is N-glycosylation, is it major in number or minor in number? The number of proteins in your bloodstream right now that are N-glycosylated or on your membranes, are they major in number or minor in number? Is this N-glycosylation, is it major or is it minor? It's major. It's major. Virtually all proteins in your bloodstream, all the blood clotting factors, all the immunoglobulins and so on, all those other enzymes and so on, like transferrin, ceruloplasmin, all those proteins, enzymes in the bloodstream, all those receptors on the plasma membranes, like the sodium pump, like the GLUT1, 2, 3, and 4, all those proteins that are in the bloodstream or in the plasma membranes are N-glycosylated. They have sugar chains in them. There are only 3 exceptions to this, but virtually all the other proteins in the bloodstream have sugar chains on them. Let me ask you this. What are 3 proteins that are not N-glycosylated? They are albumin. That's right, Luis. Albumin. What are the other 2? Albumin. What's the other 2 proteins that have no sugar chains attached to them? Other 2 proteins. Albumin's 1, Luis, correct? What's the other 2? And the other, not transferrin. Transferrin is a glycoprotein. Actin, no, actin's inside cells, not in the bloodstream. And the answer is the 2 hormone peptides, insulin and glucagon. So again, one more time. If on board exams they ask you which of the following proteins have sugar chains on them and are involved in N-glycosylation, virtually all proteins in the bloodstream and on the plasma membrane are N-glycosylated. The 3 exceptions are albumin, insulin, and glucagon. And even the other polypeptide hormones like FSH, follicle stimulating hormone, luteinizing hormone, they are glycoproteins. They have sugar chains attached to those proteins. Then let me ask you this. Why do you have sugar chains on proteins? Why are the proteins in your bloodstream, for the most part, decorated with sugars? Why? Let me ask you, why? Yeah, they have sugars on them. So what? Why do they have sugars on them? Let me see. Anybody? Why do they have sugar? Why? Energy? No. Good try. No. Nothing. No. Nothing to do with degradation. And the answer, it could have something to do with solubility, Sanap. And the answer is even more simple. That's your blood groups. Remember in hematology, the A, B, O blood groups. Remember all that stuff? The A, B, O blood group. And then you also have the other blood groups like the Lewis and Lewis X blood groups and so on. Those blood groups in hematology, those are sugar chains on proteins and lipids. And they determine, that's why I cannot give you my blood in a transfusion unless I see what is the sugar chains. Is it A, B, O blood group? So again, this is not trivia. This is major concept here. Major concept here. Major stuff here. Okay, now, okay, so proteins are going to be secreted or membrane bound. They will be, for the most part, will be N-glycosylated. Now, in the Golgi apparatus, other sugars can be added, and that's called O-glycosylation. So again, O-glycosylation occurs in the Golgi apparatus, and that's post-translational modification. After the protein is being made, sugar chains are added. So anything happening in the Golgi apparatus is post-translational because the protein's finished being made. But now it's being modified with sugars in the Golgi apparatus, and that's O-glycosylation. Now, what does O-glycosylation means? Sugar chains, they're added onto the hydroxyl group of serine or threonine. Now, later on, I'll show you the structure of serine and threonine. You will see hydroxyl groups, and so on proteins, they have sugar chains on them, and on the serine or threonine, that means O-glycosylation, and that means post-translational modification. All right, now another item. Get ready for a very high yield item here. So any protein that's going to be secreted or membrane bound, that will be N-glycosylated, and some proteins will be O-glycosylated, and some proteins will be both N-glycosylated and O-glycosylated. Immunoglobulins, for example, are both N-glycosylated and O-glycosylated. Other proteins are solely N-glycosylated, like transferrin. Other proteins are only O-glycosylated, like heparin is only O-glycosylated, only sugar chains on serine. Don't worry about the details. I doubt they're going to ask you. But now the proteins they're destined to go into the lysosomes. So here is a lysosome. So now the question is, how do the proteins that are destined to go into the lysosomes, how do they know to go into the lysosomes? There's no traffic cop that sits in the Golgi apparatus that says to these proteins, oh, okay, proteins, you now go, you get your butt over there in the lysosomes because you belong in the lysosomes. No. How does it happen biochemically? What happens is, in the Golgi apparatus, so this is post-translational modification, for any protein to go into the lysosomes, there has to be a zip code. And the zip code is applied by a very important enzyme in the Golgi apparatus called the phosphotransferase. Now, what does this enzyme do? Phosphotransferase. It's a little bit more complicated than what I'm describing, but I'm trying to simplify. What happens is, for a protein to go into the lysosomes, must have a zip code, and the zip code is applied by the phosphotransferase. What happens is, on certain sugars called mannose. So in N-glycosylation, one of the sugars is mannose, and the phosphotransferase will put phosphate onto the mannose. Specifically, it's called mannose 6-phosphate. Don't worry about the details. Just try to follow me here. Follow me. Just try to understand the concept. So one more time. So for a protein to go into the lysosomes, it must have a zip code. And the zip code is phosphate on mannose. And where does the phosphate put onto the mannose? It's put onto the Golgi apparatus by the phosphotransferase. So if the phosphotransferase is working, it puts phosphate on the mannose, and that's the zip code. And then there's a receptor that sees the phosphate onto the mannose. So here comes all these proteins, glycoproteins, moving from the ER into the Golgi and going to be secreted. And the receptor is looking at all these proteins moving through into the Golgi apparatus. And when there's a phosphate on the mannose, the receptor says to those enzymes, come into my house. My house is the lysosome. And sure enough, the receptor binds them and gets those enzymes into the lysosomes. That's how they get there in the first place. So all lysosomal enzymes are glycoproteins. All lysosomal enzymes undergo N-glycosylation. All lysosomal enzymes have the phosphate on the mannose, the zip code that gets those enzymes into the lysosomes. All right, now, if you understand that biochemistry, please, I hope you do. It's not hard. I made it simplified here. But now, if you're following me, here comes the pathology on the heels of the biochemistry. I told you earlier, any gene can be mutated. Any gene can be mutated. And sure enough, there are infants in this world who have there's 7 billion people in this world. So you got every gene can be mutated. And there are infants in this world that the gene for the phosphotransferase is genetically mutated by missense mutations. Let me see your answers here. What's the name of the disease in which the gene for the phosphotransferase is defective? Not myelin, no. That's right, it's called I-cell disease. And I-cell disease, it's where the gene for the phosphotransferase is defective. Now let me stress, in I-cell disease, some infants, the enzyme is normal, but those infants have I-cell disease because the receptor gene can be defective. So either the receptor can be defective, that's the rare form of the disease, but most often I-cell disease is due to the defect in the phosphotransferase. Now this disease, I-cell disease, you can memorize the clinical manifestations. You can memorize it, but if you think about it, it makes sense. Now let me say something here. The way I try to understand pathology, because after all, pathology is something going wrong with the biochemistry. The way I try to learn, not memorize, the way I try to learn and understand pathology is I always ask, what's the hallmark of the disease? You know, it's like some diseases, when Dr. Barone is here in a couple of weeks, It starts talking about all these diseases, and many diseases have a whole list of clinical manifestations. Like for example, diseases like Prader-Willi has a whole list, 12 or 13 different kinds of clinical manifestations. Me personally, I can't remember all that stuff. All the different clinical manifestations. For me, because I'm a simple guy, I try to ask, what's the hallmark of the disease? Every disease has a hallmark. Like for example, here I'll give you an example here of a hallmark of a disease. Everybody's going to give the right answer. Watch, here it comes. If I said to you, you see a clinical vignette, in a clinical vignette, they say the patient has the cherry red spot in the macula. What disease does that patient have? Let me see your answer here. The cherry red spot in the macula. Everybody's going to say Tay-Sachs disease, right? The number one hallmark of the disease. Now, but the problem is some other diseases may have the same hallmark. So the way I always understand pathology is I ask, what's the hallmark of the disease? And then I ask another question, what's the second hallmark of the disease, just in case another disease has the same hallmark? Because after all, what other disease has the cherry red spot? Let me see your answer. What other disease has the cherry? That's right, Niemann-Pick. So on board exams, if you see cherry red spot, you think Tay-Sachs, but it could be Niemann-Pick disease. So what's the second hallmark that says to you it's Tay-Sachs disease, not Niemann-Pick? What's the characteristic there? And the answer is the psychomotor retardation, the failure of the infant to reach motor standards. That's characteristic of Tay-Sachs disease. Cherry red spot and the psychomotor retardation. Whereas, if it's Niemann-Pick disease, we'll talk about next week. Don't worry about it now. But in Niemann-Pick disease, you'll see the cherry red spot, and the other hallmark is they have zebra bodies. Yes, they have hepatosplenomegaly. Yes. Early death. Yes. But the other hallmark is the bubbly appearance of the macrophages. The foamy macrophages is diagnostic for Niemann-Pick. So that's what I'm getting at here is. So every disease in pathology, in my opinion, easy way, what's the hallmark of the disease? And what is a second hallmark in case another disease sounds very similar? Okay. Now here you see a perfect illustration of this with eye-cell disease. Now eye-cell disease has two hallmarks. I could tell you the answers right now and move on, and you'll forget about it tonight because you're trying to memorize. I'd rather have you figure it out first. Here. All right, now just picture. Now again, if you have the answer in front of you because you wrote it down, you watched the videos, please don't answer. Give everybody a chance. Okay, now think about this a minute. In eye-cell disease, suppose the phosphotransferase is defective. Then in eye-cell disease, here comes the question. Are those enzymes, those lysosomal enzymes don't get the zip code, but are they still, yes, I'm looking for a yes or no. Are they still, are the lysosomal enzymes still being made in the ER network? Yes or no? In eye-cell disease, if the phosphotransferase is defective, are the lysosomal enzymes still being made in the ER network? Yes, that's right. So they're still being made on the ribosome, and then they finish getting made in the ER, and then they go into the Golgi apparatus. But now think, if the phosphotransferase is defective, you don't put the phosphate on mannoses. But now what happens to those hydrolytic enzymes? Do they hang up in the Golgi apparatus, forming an inclusion body? Yes or no? So one more time. The lysosomal enzymes, they get into the Golgi, but they don't get the phosphate on the mannose. Do they form an inclusion body? And the answer is no. They don't form the inclusion bodies. So what happens to those enzymes then? Now just picture, they come into the Golgi. If you don't put the phosphate on the mannose, what happens to those enzymes? They get secreted into the bloodstream where they don't belong. Now think. So all these hydrolytic degradative enzymes, they're supposed to be in the lysosomes. now are secreted into the bloodstream where they don't belong. Now just picture what's going to happen. In these infants, tragic disorder. If you ever see a patient with eye cell disease, tragic. But in any case, what happens is in the bloodstream are all these hydrolytic enzymes. What's going to happen in the bloodstream? These hydrolytic enzymes, degradative enzymes, slowly but surely, even though it's slowly because the pH is not the correct pH, but anyway, slowly but surely, they're going to start breaking down, breaking things in the vasculature. You think the body likes that when things are breaking down in the vasculature, the bloodstream? New epitopes are being generated because things are breaking down. How does the body respond? What is the pathology, one of the pathologies associated with eye cell disease? Just picture. Okay, you can now answer the question. So when things are breaking down in the bloodstream, new epitopes are being generated, how does the body react to that? How does the body deal with that? And the answer is, here comes one last chance. That's right, Dave, that's right. Things are breaking down, and one of the characteristics, here it is. There it is. Generalized inflammation, not acute inflammation that you would see in gout. This would be generalized inflammation. Things are breaking down, new epitopes are being generated, the immune system kicks in, generalized inflammation. It must be so painful in these kids. Generalized inflammation, one of the hallmarks. It makes sense, right? You don't have to memorize this. It makes sense. Generalized inflammation. You know, at my institution, every year when I lecture to my medical students, every year at the end of the course, the students write evaluations of me. You're going to be writing evaluations of me as well, okay? But my students wrote evaluations of me. And a few years ago at my institution, when I read the evaluations, there was a medical student wrote, well, maybe, because I use that phrase all the time to my students, it makes sense. You know, this makes sense. It makes sense. So at the end of the year, when the students wrote an evaluation of me, there was a medical student wrote, well, maybe it makes sense to you, but it doesn't to me. Man, I wish I knew who that student was. I'd like to talk to that student. If it doesn't make sense, find out why. Don't make excuses. If it doesn't make sense, find out why. Read about it, ask me, ask a colleague, but find out why. You must know why. Things make sense. The national board does not want to hear excuses. If you say, oh, I don't know why it doesn't make sense, you're making excuses. Find out why. The national board doesn't want to hear excuses. They don't want to hear, I hate to be cruel here. I hate to be harsh, but the boards don't want excuses. Oh, I had biochemistry 8 years ago. Oh, and biochemistry was not taught properly in my institution. That's excuses. They don't want to hear excuses. They want to know, do you know the answer or not? So the idea is make sense of things. It makes sense. They have generalized inflammation. Now, the other hallmark of the disease, a lot of things cause generalized inflammation, but the other hallmark of the disease also makes sense if you think about it, not memorize. All right, now the idea is, what does the I and eye cell stand for? Let me see your answers here. What does the I and eye cell stand for? That's right, it stands for inclusion bodies. Okay, so now they do have inclusion bodies, but not because the enzymes are causing the inclusion bodies, but why do they have inclusion bodies? They have a multitude of inclusion bodies. And the answer becomes simple if you think about it. So just picture a macrophage from an eye cell patient engulfs a microbe and kills the microbe by the respiratory burst. And then in the cell is a dead microbe. And the dead microbe is supposed to be broken down in the lysosomes and so on. But now in the lysosomes, it's empty. There are no enzymes in the lysosomes because they're out in the blood where they don't belong. So now what happens to that dead microbe? It just sits there and sits there and sits there. Also in other cells, fibroblasts, liver cells, all receptors. transferrin receptor, LDL receptor, bringing things out of the blood into the endosome, which fuses with a lysosome, and that LDL is supposed to be broken down. Transferrin broken down by proteases in the lysosomes, but the lysosome is empty. So what happens to the LDL? What happens to the transferrin? It just sits there and sits there in the lysosomes. So there it is. What are in the inclusions? And the answer is simple, because you think about it. Food gets in, but can't be degraded. It's a mixed bag of junk. All kind of stuff coming into the cell, but can't be degraded. It just makes sense. So let me stress here. On board exams, they love inclusion body diseases. And so later on next week, we're going to get into this sphingolipidosis. And that's an inclusion body disease. And so is eye cell disease. And the boards love it when two or more diseases sound very similar. They're going to want to know, how do you know which disease does this child have? Is it eye cell? Is it Niemann-Pick? Is it Gaucher's? Which, if you see inclusion bodies. And next week I'll get into the issue, the differential diagnosis. But notice here with eye cell disease, it's a mixed bag. It's not one kind of molecule accumulating. It's a mixed bag of junk accumulating in the cell. It makes sense. And if it doesn't make sense, find out why it doesn't make sense. Don't make excuses. Find out why. All right. Now, what's coming up next is the last of the high yield figures, and that is the all important topic of collagen. Now, you might ask, why are we talking about collagen now since we're in molecular biology? And the reason is because collagen is often, besides all the pathologies associated with collagen, collagen is often used as a major illustrator of how a protein undergoes co- and post-translational modification. How a protein, while it's being made and after it's being made, how it could be modified. So collagen, out of 322 questions, at least one of them or more will likely be a collagen question. You must know collagen, not only for the pathologies, which will come up on this figure, but you also need to know the basic modifications that can go on. All right, now the idea is, what is collagen biochemically? And what collagen is, it's three peptides wrapped around one another. And in each of the three peptides, it has the sequence glycine XY. XY can be any amino acid, but often it is hydroxyprotein, but every third amino acid is glycine. Now let me ask you the why question. Why? See, let me see your answers here. Why is every third amino acid in collagen glycine? Do you think it just happens to be that way, or is there a reason why? To make it packed, tight, small. That's right. There we go. That's right. And the reason is because we'll talk about the amino acid structures later on, but glycine has the smallest R group, the smallest side chain. And because it has a small side chain, it's easy to wrap three peptides around each other, forming the triple helix. There's no steric hindrance. But if every third amino acid were valine or isoleucine with big bulky groups, you would never form the triple helix. That's why every third amino acid must be glycine. Now let me ask you another question. In histology, what cell type makes collagen? Let me see your answer here. What cell type makes collagen? What cell type makes collagen? That's right. It is the fibroblasts only. No exceptions. So the fibroblast makes collagen only. So what you're looking at on page 64, suppose this represents a fibroblast, And in the middle part, in the blue, that represents the Golgi apparatus, because after all, each of the collagen peptides will be secreted. So the protein, as you might imagine, so what you're looking at here is this is the Golgi network, and these little two circles represents the ribosome. That line represents one of the three collagen peptides. The wavy part represents the N-terminal hydrophobic signal sequence, because they're going to be secreted. They must have the N-terminal hydrophobic signal sequence. All right, now before we move on, what's the name? Everybody should get this. What's the name of the disease in which one of the collagen genes can be mutated? What's the name of the disease in pathology? Everybody should get this. Not Marfan. No, not Marfan. That's a different protein. That's fibrillin. That's right. It's called osteogenesis imperfecta. Now, which collagen? Now, you know, OI is characterized by the two hallmarks: easy fracturing. A lot of things cause easy fracturing. For example, vitamin D toxicity causes easy fracturing. So, but OI causes easy fracturing. What's the other hallmark of OI? Blue sclera. You see those two words on board exams? Piece of cake. They're talking about OI. Now, which collagen is it? You must know, high yield, which collagen type is responsible for OI. That's right. It is collagen type 1, because that's the collagen you find in skeletal tissues. All right. Now, what in pathology? What is another pathology that's associated with collagen gene? A collagen gene. Which one? Not Ehlers-Danlos per se. No, it is. I'll talk about Ehlers-Danlos in a minute, and that is Alport syndrome. Now, Alport syndrome. What is the collagen type there? What's the collagen type there? That's right. Yeah, well done. That collagen is collagen type 4. That's the one you find in the basement membranes. So that's the aneurysm issues and so on, but that's the basement membrane collagen, not in the skeletal tissue. All right, now let's high yield. Let's proceed. So then, so coming back to the normal situation. So then the N-terminal hydrophobic signal sequence may be cleaved off. It served its purpose. So in this case here, it's cleaved off. It gets the protein into the ER, and while it's being made, now we've got an arrow coming up here. There are a pair of enzymes, two enzymes. One of the enzymes hydroxylates protein residues. Another enzyme hydroxylates lysine residues. The name of the enzyme, not shown here. The name of the enzyme hydroxylates protein. What's the name of that enzyme again? Okay, I'm drawing a blank. The name of the enzyme that hydroxylates protein. Oh yeah, protein hydroxylase. Wow, biochemistry is so hard sometimes. Uh-oh. The name of the enzyme hydroxylates lysine residues. Uh-oh. Name of the enzyme hydroxylates lysine. What was that enzyme again? Oh yeah. Lysyl hydroxylase hydroxylates lysine amino acids on the collagen. Wow, so hard sometimes, man. Any case, now this arrow right here might not work for two reasons. One reason is genetics, and when the protein hydroxylase is defective, that gives rise to Ehlers-Danlos 6. So now there's many different kinds of Ehlers-Danlos. Dr. Barone will talk about the other ones as well. But Ehlers-Danlos 6 is when the protein hydroxylase is defective. Now this arrow might not work for two reasons. One is genetics, something wrong with the gene. The other reason why this arrow won't work is nutrition. And when this arrow does not work because of nutritional reason, what's the name of the pathology? Vitamin C deficient. That's right, it's scurvy. So in scurvy and Ehlers-Danlos, this arrow won't work. Now let me ask you this. In scurvy and Ehlers-Danlos, it has two hallmarks. Those diseases have two hallmarks of the disease. Many clinical manifestations in Ehlers-Danlos as well as scurvy. But the two main hallmarks, what are they? Let me see your answers here. What is the hallmarks of scurvy? Yeah, they have hemorrhaging, bleeding gums. Now, we'll talk about that issue later on. Don't worry about it now. We'll talk about why is it in scurvy you bleed, hemorrhage, bleeding gums. But what's the other hallmark of scurvy and Ehlers-Danlos? Because that's what we're going to talk about here. What's the other hallmark of scurvy and Ehlers-Danlos? What about the skin? What about the Marlon? What about the skin? You're on the right track. Maritza, there we go. That's right. The hyperelastic skin. If I had scurvy, I could take my skin and stretch it. That is the hyperelastic skin. How do you explain that in scurvy? You could memorize that. How do you explain it? And it's not hard. It's not hard as long as you think. And okay, here comes the explanation. So we're trying to explain one of the two hallmarks of scurvy. Why do you have hyperelastic skin? Why do you have hemorrhaging? We'll talk about later on. But the idea is, why do you have a hyperelastic skin? And the reason is, when you make collagen, collagen is elastic. If this arrow is working with the hydroxyl groups, collagen still is elastic. So whether you put the hydroxyl groups on collagen or not, collagen is elastic. What happens, however, there is an enzyme coming up in a minute or so that works downstream that must see the hydroxyl groups on the lysine residues. And when the hydroxyl groups are on lysine, the enzyme downstream is responsible for making collagen rigid. So if this arrow does not work because of scurvy or Ehlers-Danlos, you don't make hydroxylysines, the enzyme downstream never gets a chance to work, and collagen remains elastic. Okay, one more time. I know I covered a lot of ground here. So when you make collagen, it's elastic. And if this arrow works, hydroxylation of protein and lysine, collagen is still elastic. But now if there's hydroxyl groups on lysine, there's an enzyme coming up downstream that makes collagen rigid. And we'll talk about what's it doing to make collagen rigid in just a minute, a couple minutes here. All right, now let's proceed. Again, understanding the biochemistry explains the pathology. Let's proceed. Coming back to the normal situation. When there's hydroxyl groups on lysines and prolines, some of the hydroxylysines get sugar chains added, and then afterwards inside the cell we get the triple helix formation. Again, and then what happens is the protein becomes secreted. The collagen triple helix becomes secreted. Outside the cell, now this is post-translational modification. After the protein is secreted, the N-terminal and C-terminal peptides are cleaved off. The middle part is called tropocollagen. So the N-terminal, C-terminal peptides are cleaved off by specific proteases. You don't have to know. The middle part is tropocollagen. And then what happens is the tropocollagen self-assembles, forming the collagen fibril. The tropocollagen binds to each other, forming the collagen fibril. And it's still elastic. So the collagen fibril is still elastic. And then this all-important enzyme called lysooxidase, which requires copper. This is the enzyme that's responsible for making collagen rigid. Let me see your answers. What does the enzyme do to make collagen rigid? Let me see your answers here. What is the enzyme doing to make collagen rigid? Look at the name of the enzyme. It's helping you out. Lysooxidase, requiring copper. Anytime you see a heavy metal ion, it's referring to electrons are being involved. Most likely some sort of oxidation reduction reaction going on. And sure enough, what the enzyme does, It's oxidizing hydroxylysines to form these aldehyde groups that you don't have to know. The aldehyde groups then cross-link with each other, and that's what makes collagen rigid. So one more time. Lysooxidation in the presence of copper oxidizes the collagen to make collagen rigid by cross-linking that goes on. That's why collagen is rigid, because of cross-linking. You must know the biochemistry of what goes on, not the gory details. It's the straightforward concept of the biochemistry. You must master why, when the steroid doesn't work, collagen is hyperelastic. If you understand that biochemistry, it goes a long way. Now, what we're going to do is we're done with chapter 4. We're going to do 2 sample questions. We've got a few minutes to do the sample questions here. So, if George can bring up the selection pod. Oh, by the way, Menkes disease, but I forgot to mention this there. Menkes disease is when you're missing copper. So in Menkes disease, the kinky hair syndrome, where collagen is not made properly, is defective, it's because of the copper problem. So Menkes is a copper problem, which this enzyme needs, lysooxidase. All right, so what we're going to do here is I'm going to do 2 questions here, 2 vignettes, and then we're done for the day. And the first question comes from your book. Don't look at the answer, but it's a difficult question. Students always ask me about this question, so I'm being proactive. I'm going to show it here. And it looks very formidable, but it's an example question that you might see on board exams. So here, and if George, if you can bring up the timer and set it for 1 minute, here comes the question. The 2 clinical vignettes. The first question, 1 minute. Here it is. Let me move this out of the way. I don't know where else to put this. Maybe we'll move it over here. Okay. 1 minute. Can everybody see it? Okay, move this over here. Okay, there we go. There we go. Thank you. Ten seconds. Okay, and the minute's up, and if it's end the poll and broadcast the results, and looks like, man, numbers are all over the place. Wow, that's why I'm doing this question, because I know it's a nice thing about being a veteran. I can tell when students are going to have a question. Now, this is by the way, this is the real life situation that goes on in cystic fibrosis. We know what the mutation is. There's a 3 nucleotide deletion in exon 10 of the CFTR gene. Now, what you're looking at here in the top part, these are the codons for the normal gene at position 506, 507, 508, and so on. The first 505 codons are not even shown here. So the way to do this is, if you're given the genetic code, look at these codons and ask, what's the amino acid specified by that DNA codon? So ATC start at position 507. ATC in DNA means AUC in RNA. AUC, here it is, AUC in RNA means isoleucine. So let's draw it down. So by the way, the answer is B here. The answer is B. So here's the isoleucine. So ATC in DNA means AUC in RNA. AUC in RNA is isoleucine. TTT in DNA means UUU in RNA. That means phenylalanine. GGT in DNA means GGU in RNA, And GGU, here it is, GGU in RNA, that's glycine. And next one is valine, and there's serine. So the way to do this is you first figure out what is the amino acid sequence that's normal. There it is at position 507, 8, 9, and 10. And then because of the 3 base deletion, that's what the 3 dots are about. By the 3 base deletion, the T to third position at position 508 now goes to the third position at 507. These nucleotides on the right side now move by 3 positions from your right to your left because of 3 base deletion. So now all you got to do is draw the new nucleotide sequence in the mutant gene. So at position 507, what's the codon at position 507? And the answer is it will be ATT, because this T at the third position at position 508 now goes to 507. And then the GGT goes to position 508. So in other words, all the nucleotides are shifting from your right to your left by 3 nucleotide positions. So this is now the new sequence of the mutant gene. Then all you got to do is look at the genetic code and figure out the amino acid sequence based on this mutant sequence. So here it comes. ATT in DNA means AUU in RNA. Here's the AUU in RNA. That means isoleucine. So let's draw it down. GGT in DNA means GGU in RNA. There it is. There is the glycine. Next amino acid is valine, and that's serine. Look at this sequence of amino acid sequence. Look at this sequence, amino acid. What's the difference between the normal and the mutant sequence? And there it is, option B. Deletion of phenylalanine with no change in the C-terminal sequence. The C-terminal sequence of the normal gene is glycine, valine, serine. The mutant sequence is glycine, valine, serine. Deletion of phenylalanine at position 508. That's the normal, that is the actual real life situation. Deletion of phenylalanine at position 508 with no change in the C-terminal sequence. Yes, I know it's a difficult question, and you're saying, oh man, I only have 78 seconds per question. You have to think quickly. You can't dawdle here. You have to think how to approach these questions here. It's not a hard question. It's not tricky. It's a matter of thinking your way through it. All right, now we'll do one more question, then we're done. But the next question is not as difficult, and the question that I wrote. So here comes the next question. Last question, then we're done for the day. Okay, here it is. 1 minute. Ten seconds. Okay, it's closed the poll and broadcast the results, and half the students put option D. A lot of students put option E, and the answer to this question is option D, mismatch base repair. Now notice here, notice here, they're not asking what's the name of the pathology. Let me see your answers here. What's the name of the pathology this woman has? What's her problem? What's the name of the pathology? That's right. It's called hereditary non-polyposis colorectal cancer, also called, a lot of people got it as well, Lynch syndrome. Now, how do you know it's Lynch syndrome? Because they're saying to you in the vignette that a brother has colon cancer, a maternal uncle's death in the 50s from a similar carcinoma. That's a major clue. On board exams, if they tell you about a sibling, a relative, something in the family, another person in the family has the same disease, it's telling you something. It's telling you this is a genetic disease, not a nutritional disease like vitamin C deficiency, not an environmental disease or problem like lead poisoning. If I talk to you about other people in the family having the same disease, you automatically think they're asking me about a genetic disorder. Major clue. All right, now, this woman has a colonoscopy and abnormal growth in the colon, but they're not asking what's the name of the pathology. They're saying a gene that deals with mutations in DNA. Which of the following mechanisms is deficient in this patient? Now, without looking at the previous figures or chapters, what's the name of the gene or genes when they're defective gives rise to Lynch syndrome? Let me see your answers here without looking. What's the name of the genes? They could ask that. They can say, which of the following genes might be mutated gives rise to this patient's problem? That's right. And the answer is, it's HMLH1 or HMSH2. Remember, one of these genes encodes for a protein which finds a mismatch base and the other takes out the problem. Now let's go through the other options here, and then we're done. Pyrimidine base excision repair. What did we talk about that is involved in? Look at the words. Pyrimidine base excision repair. Not thymine dimer. No, not xeroderma pigmentosum. No. Look at the words. Pyrimidine base, not bases, not plural. Pyrimidine base, that is, remember, cytosine deamination. Remember, nitrates convert cytosine to uracil. A pyrimidine base excision repair. Option B, what disease that we talked about is associated with option B? Direct repair of pyrimidine dimers. What disease? That is the xeroderma pigmentosum, and the name of the enzyme was the excision endonuclease, the direct repair of pyrimidine dimers. Now, option C, 5' exonuclease nucleotide removal. Where did we talk about that, and what process or whatever specific process was the option C involved in? Just trying to get the recall. Where did we talk about option C? That's right. Well done, Efosa. That's right. Hayal, that's right. That is the removal of the RNA primer was the 5' exonuclease, not the 3'. The 3' exonuclease was the proofreading enzyme. And lastly, option E, inability to carry out apoptosis due to a tumor suppressor gene defect. What protein or gene that did we talk about in this class that deals with option E? That's right. That was the disease was Li-Fraumeni syndrome. The name of the gene was P53. Here. Okay, so again, now we're done for the day. Now tomorrow we're done with chapter 4 today, and tomorrow we'll do chapter 5, major chapter on gene expression, major, a lot of integration with all disciplines coming up in chapter 5. You want to be here in the first hour. And then after that, we're going to get into cloning issues, and then we'll start to get into genetic testing. So all that basic molecular biology has been covered. What's coming up in chapters 5, 6, and 7 is modern molecular biology, how you analyze genes, how you do gene expression, modern molecular biology coming up tomorrow. And now when we're done today, I'm going to post on the Facebook 3 more questions. Now, when I looked at today's Facebook, only about a half a dozen students or whatever are answering. Please, and go to the Facebook and look at the questions. It's for your benefit, not mine. It's for your benefit. So please look at the Facebook, give answers, and I'll give you the answers tomorrow. Now I forgot my email. So a lot of you didn't message me on Facebook, so I'll give you my work email. It's this, my last name, very easy. It's Turco, very simple. Turco at uky.edu. UKY for the University of Kentucky. .edu for education. So Turco at uky.edu. So we're done for the day. I want to thank George and Dr. Donald and Dr. Seda for all the help they've given us tonight. as assistants. Very, very helpful people. So thank you so much. So everybody, so again, hopefully you're learning. You're not memorizing, but you're trying to understand. You're making sense of this discipline called biochemistry. No reason to fear it. Understand it. Make it your friend. Okay, everybody, we're done for the day, and I'm going to go off camera and see you tomorrow. All the best. Have a safe evening.